So good morning, everyone. We're right at 8 o'clock Eastern Time, so we're going to get started this morning. We just wanted to welcome everyone to day 2 to the HESI webinar on benchmark dose modeling for risk assessment, building a path toward harmonization. We had over 1,200 people register for the webinar and are really pleased to have such a diverse cross-sector audience joining us today. I'm going to introduce Andy Nong from Health Canada, who is going to be giving us the introduction and the overview of what we can expect from today. So thank you all, and take it over, Andy.
Thank you, Claire. Just a few welcome everybody. I'd like to start out with some ground rules. So make sure you're muted. Please answer your questions in the Q&A today. Please be concise and clear when you write your questions. Be respectful as well. We ask you all to upvote any question of interest so we can answer them as possible and follow up if needed. If you have any technical issues, please email at the email below you have here. Next slide. So I'd like to welcome you. In terms of there's currently an introductory poll that we ask you all to follow. Please use the QR code or go to the website and enter the code below. This is very informative for us and be able to learn more about you and everybody else that's been attending and help us in terms of our engagement. Next. So on behalf of the committee, I'd like to welcome you all to today. Yesterday, we're on today's agenda for day 2. Yesterday, we had very, very great speakers, and I'd like to follow up with another series of speakers. We learned about BMD, we heard about how they're being applied, and we learned how they're used for guide studies and so on. Today, we'll be following up with the regulatory speakers and how they're used in context, and then going in deeper dive in terms of the application in different areas and uses. So this morning we going to start off with our some speakers regarding some the EPA benchmark dose guidance, following some EFSA presentation from EFSA, and then we'll go in deep dive and, like I said, in case examples in agriculturals and industrials and so on. So I'd like to go to the next slide. Thank you. And here's our committee. We have a great group of people here that's been helping out and putting together these 2 days workshop. And we hope to be able to hear more from you, and they're expert in their areas, and be glad to answer any questions as well as our speakers today. Next. So why we have this workshop? We're seeing an increased use of BMD modeling and risk assessment in different sectors. I think that's been major areas that it's not concentrated in one area. It's all over right now being used. But in the application, we're seeing a lot of challenges, and we hope to be able to harmonize this and understand across the areas. So it's multiple tools and approaches. There's challenge and differences in statistical methods, model selection, and BMR choices, priors and fittings, and complexity across different diverse data types. So we saw a lot of questions regarding BMR data types and studies and how they were being used. And there's an ongoing tension between statistical rigor and biological relevance. So what is my best approach to using the data? How should I be setting up my BMD modeling approaches? So there's some inconsistencies in the application across the sectors and agency, and we're going to be able to have an understanding how it's being used today and how we can make it a better approach for all to understand. So next. So our goals really to convene regulatory and academic and industry experts, share the experience and methodology, support broader adoption of the BMD and risk assessment, identify key challenges and opportunities for harmonization. And hopefully after this webinar, we'll be able to build a collaborative cross-sector network within HESI and develop scientific, defensible, consistent approaches. And then we'll be able to lay the groundwork for future collaboration, working groups, and follow-on activities. So day 1, we saw a lot of our goal was really to build and understand. So we saw the basic building block of how BMDs are used and how they're being applied. We saw differences in the approach, the barriers that are in the areas. So we compare some perspectives of how they're being used in different areas and learn the outcome in terms of priority and challenges. Today, we're going to go in deeper dive. We're going to translate insights into harmonization pathways, examine how regulatory application of BMDs, and then look at the regulatory frameworks and their case studies and use in different sectors and diverse, and then identify where there's approaches that align or diverge. And we hope be able to have an idea of the barriers and ways and enablers to harmonize and evaluate feasibility and aligning in terms of model selection and approaches and BMR choices, data interpretation, and so on. So at the end, we hope to be able to see some actionable areas for harmonization. We saw from the poll yesterday that harmonization is there's a consensus we want to harmonize, and there's this possibility that we can have areas where we could be able to prepare for future working groups and inform future collaborations. So I'd like to go to the next slide, which is passing on to Claire to start off our presentations today. Thank you.
Thanks so much, Andy. So with that, we're going to get started on our first session, which is regulatory guidances on BMD modeling. And I'm happy to introduce our first speaker, who is Alan Davis from the US EPA, who will be talking on the US EPA guidance on BMD modeling.
Yeah, so thanks for inviting me to give a presentation on EPA's guidance on benchmark dose modeling. It's yesterday's presentations were interesting, and it's always interesting to see where the field of dose response is heading, and I think there's a sense that the BMD guidance is a bit out of date at EPA, and I kind of want to maybe acknowledge that a little bit, but also dissuade that opinion as well. So
Alan, can you try to turn your video on really quick? I had turned it off earlier.
Sure.
You should be able to. I'm sorry about that.
No, you're fine. Start my video. Cool. Hello. Okay, so standard disclaimer, if I say anything correct, it is reflective of the views of EPA. If I say anything wrong, it's reflective of my views. So, benchmark dose guidance at the EPA. There are a number of resources for BMD guidance at the EPA. The number one-stop shop is the BMD technical guidance document. There are other ancillary guidance documents relevant to BMD modeling that mainly reflect how to model certain types of data, developmental tox, repro tox, genetic tox, and then how other quantitative methods like PBPK modeling or default dosimetry intersect with BMD modeling. And then there are other guidance documents that are filtered through the lens of like how the guidance is applied in BMDS, like the BMDS user guides and technical memos, etc. So the 800-pound gorilla in the room is that the BMD guidance was first released in 2000. It was reflective of recommendations made at a 1995 workshop. And then following a torturous and very long review process, it was finally released in 2012. So, at this point, it's almost 15 years old. It does cover multiple issues that are, absolutely 100% relevant to BMD modeling today, including data susceptibility, benchmark dose response selection, and then some, aspects of modeling that the field of dose response has moved past, or additional more robust methods have been developed and have started to be applied. And so issues related to model fitting, model selection. But the people that wrote the BMD guidance were very smart, and they knew that the field of dose response was burgeoning. It was going to continue at an increasingly rapid pace, and it was very likely to outstrip the ability of EPA to release timely guidance. And so they put in probably what I think is the most important statement in the entire technical guidance in section 1.1, where it just says, the guidance reflects current knowledge, understanding, and experience, but the document's intended to be updated. Unfortunately, that really hasn't happened. But The document should not be viewed as precluding research into quantitative risk assessment methods or adoption of those approaches in human health risk assessments. And so, people like to think of the somewhat incorrectly that the technical guidance is prescriptive. It lays out how a dose response analysis has to happen. You will always follow these steps and your decisions will always be the same. And that's not really the case. And instead, this statement really makes the technical guidance permissive. It does make recommendations, but throughout the document, it provides counterexamples. Hey, we recommend this, but this method is sort of gaining prominence, and you might, view it as an alternative approach. And so the technical guidance really sets a general framework on how people should think about dose response, and just identifies, topics and decisions that people are going to have to make. And, it's sort of glib, but my colleague, Jeff Gift, my mentor, used to say in our benchmark dose training sessions that the correct answer is whatever you get through peer review. And that's kind of, off the cuff, but it's true. And the technical guidance really, instead of saying, this is how you do a dose response analysis, it sort of just provides that general framework for you to build the rationale and justify the dose response decisions that you've made in your analysis. So, there's a very extensive discussion on data evaluation. So, the first step in dose response is selecting the data that you are going to model. And, as it was pointed out yesterday, you can't model every endpoint that is in your database unless you're modeling a very limited database. You're going to end up modeling endpoints that aren't relevant, endpoints that aren't going to drive the decisions in your risk assessment. So you need a approach to sort of filter out the data that you move into dose response. And some of that's going to be toxicological, some of it's not going to be in the dose response range, but there are data quality issues that will absolutely impact what endpoints you end up modeling. And so the BMD guidance provides this rough framework and steps through, a number of questions that you need to answer. First, are there sufficient data? Different types of data require different amounts of information to adequately model. Dichotomous data only requires dose, number of subjects, and incidence or percent affected. Continuous data, you need either the individual responses for the animals, or you need summary measures of the mean response and some measure of variability. If you don't have that data, you can't model it. Other questions are, is there a trend in your data? The technical guidance makes it clear that statistical significance is not required. If you have, a monotonic trend in a rare endpoint, you can make a justification that the response that you're seeing is biologically significant and warrants modeling. Are there enough dose groups in your in the data you want to model? And as it was discussed yesterday, we prefer studies with more dose groups and monotonic responses. There is a interplay in between the number of dose groups you have and the total number of subjects, or the number of subjects per dose group, that you need to be aware of. Studies with only one dose showing an elevated response may not support BMD modeling. It may, if that response is near the benchmark response, but you just need to be cognizant of those patterns in dose response. And then specifically in the realm of frequentist modeling, maximum likelihood estimation methods, you want the number of dose groups to be at least as large as the number of model parameters. You don't want to try to fit a 5-parameter model to a data set that only has 3 doses. Data evaluation, if you've answered these questions and you get to a preliminary set of data sets, you need to start looking at the actual trends and patterns in your dose response data. And so, as I just spoke, a data set with only the highest dose showing a response, you don't really have any information about the shape of the dose response curve in between the highest dose and the penultimate dose. It could be sigmoidal, it could be hockey shaped, there could be a threshold. You just have limited information. If that response is much greater than your BMR, you have even less response. You have a little bit more confidence in modeling this type of data if that response is right at 10%, say if you're using an extra risk of 10%. But still, this data really does indicate a general lack of information about what the real shape of the dose response curve is in between those two doses. The converse of that is when you have elevated responses at all your dose groups that are roughly equivalent to one another. So instead of missing the shape of the dose response curve at the high dose region, you're missing the shape of the dose response curve in between control and this lowest dose. And again, the BMD is going to be bracketed between these two dose values, but you just have no information about the shape of the curve. And so this is not a data set that we would typically recommend modeling, and the technical guidance is pretty clear that you shouldn't model this type of data. Highly superlinear curves, where you do have a monotonically increasing dose response curve, but the response in the lowest dose is much higher than the BMR. Again, the design of this study has really missed the region of interest with respect to biologically relevant responses, and so typically we recommend not modeling highly superlinear curves like this. Some of the parameter constraints that Lynn discussed are relevant to if you have to model data like this, and I'll briefly go over those again later. But again, generally not the best data. The technical guidance dedicates a lot of real estate in discussions about selecting the benchmark response for dichotomous and continuous data. Lynn covered this yesterday, so I'm going to kind of breeze through this, but an extra risk is recommended as the standard for dichotomous data, customarily because it's at or near the limit of sensitivity in most cancer bioassays or non-cancer studies of a comparable size, so n's around 50. But again, given that the technical guidance is not prescriptive, it's permissive, it does spend a lot of time discussing situations where you're going to step away from this recommendation. So, situations where the use of different BMRs are supported. You can decrease the extra risk if you're modeling frank effects like seizures or death, if you're modeling precursor effects or adaptive effects that aren't necessarily adverse, you might actually increase the BMR. If you're modeling a cancer endpoint where your point of departure or the toxicity value that you're interested in is the slope value, you may increase the BMR to get it close to where the response rate is in the lowest dose. If a study has greater than usual sensitivity, you can lower the BMR because that study is going to have higher power to detect smaller responses, and so the most often used example of this is, using a BMR of 5% extra risk for developmental studies. But then technical guidance also says or recommends that you present the results for a 10% BMR for comparability across assessments or for cumulative risk assessment purposes. So this is just an example of why extra risk is preferred. If there is any response in the control group, extra risk is going to derive a lower BMD, so it's more health protective than added risk. Continuous BMR types. This is where the technical guidance really delves down into this issue, because selecting the BMR for continuous data is a little bit more nuanced than dichotomous data. The preferred approach is to select a BMR that represents some minimally biologically response level. Examples: 10% decrease in body weight, 10% increase in liver weight. If you have information that informs what percent change is biologically relevant, by all means, the technical guidance says that's the way to go. But it acknowledges that this response rate is typically or commonly going to be lacking. And so in the absence of biological considerations, it recommends a BMR change equal to 1 standard control deviation. But again, as with dichotomous data, it lays out a number of examples where the use of different BMRs is supported. The selection of what type of BMR may impact the derivation of your BMD quite a bit. And so in this example, we're looking at almost a 2-fold change in the BMD, 10% versus 1 standard deviation. And so, users just need to be cognizant of how selection of the BMR is going to impact your modeling results. Lynn covered why we use the standard deviation for the BMR for continuous data, and it's based on Kinney Crump's 1995 paper that posited an example that, if you assume that 1.4% of the animals in the control distribution are responding adversely, and you shift that that distribution by 1 standard deviation, you're going to result in approximately 10% of the animals falling below that adversity cutoff level. And that 10% is roughly analogous to the 10% extra risk that we use in dichotomous modeling. This assumes a simple shift in a normal distribution. Some toxicity responses may not behave that way, and users need to be cognizant of that. A more general approach to the standard deviation BMR is addressed in the technical guidance. It's called the hybrid approach. And this approach explicitly uses risk expressed in the same manner as with dichotomous data. So extra risk, you can use added risk if you want, but we recommend added risk. And so for decreasing responses, the BMR and increasing, the BMR are going to be equal to these 2 equations, which look somewhat complicated, but you can triangulate them to the equation for the extra risk BMR, and you can see how they're directly analogous. 2 parameters need to be selected by the user. The benchmark response that you're interested in. So this is your extra risk, 10%, 5%, whatever. And then the background rate of response. This can just be, I'm going to use 1%. You can leverage biological data here to inform this background rate. And it's something that we did in a number of the PFAS assessments where we were modeling epi data for birth weight, and we used the clinical definition of low birth weight and then the known percentage of babies in the US that fall below that background rate or that cutoff value. So we leverage biological clinical information. Sometimes that's not going to be available. And so the example here is if you consider that BMR of 10% extra risk and a background rate of 1%, you can calculate, the shift in the mean response that is going to result in 10.9% of the animals or humans falling below that cutoff value. This should look familiar to what was just shown for the standard deviation. And so the hybrid approach really is a generalization of the standard deviation BMR type. And it just gives users a little bit more flexibility in selecting the parameters that go into the calculation of the mean response of interest. Modeling data, this is where the technical guidance shows its age a little bit, but again, it provides some tantalizing hints that new methods are entirely appropriate. The BMD, the technical guidance recommends maximum likelihood estimation as the general way for parameter estimation and model fitting. The field of modeling is the broad arc of dose response modeling seems to be trending towards Bayesian methods, and the technical guidance acknowledges that Bayesian approaches for BMD estimation are valid. It doesn't provide strict guidance on the application of those approaches due to the newness of the methods at the time. But it does say Bayesian methods are okay. So, people shouldn't look at the technical guidance and go, oh, well, it says you have to use MLE methods. It doesn't. So Bayesian methods are okay. They're copacetic as far as the technical guidance is concerned. It also supports incorporating information on covariates when fitting dose response models. The recommendation is made specifically in the context of developmental tox data, like using a litter-specific covariate, but it shouldn't take too much justification to say, well, if that's okay for nested dichotomous data, it's okay for dichotomous data and continuous data as well. So the use of covariate modeling is entirely appropriate according to the technical guidance. Parameter modeling constraints. I'm not going to go over this other to say Lynn covered this yesterday, and depending on the model, the parameter constraints that you use or don't use, it's going to impact your modeling results. And the technical guidance recommends certain modeling constraints, parameter constraints, but it doesn't say these are absolutely necessary and you have to use them. It lays out a situation where users can, apply different parameter constraints, look at the modeling results, and determine, which is most appropriate. If you model data with an unrestricted Weibull model and you get a BMDL of 0, probably not the right thing to do. So if you apply that constraint and you get an actionable BMDL, that is low, but it's not 0, that might be the approach to use if you absolutely have to model this highly superlinear data. Step through this real quick. The technical guidance lays out a preferred model hierarchy. At the top, gold standard is pharmacodynamic or biologically based models when sufficient data are available, and that sufficient data are available are doing all the heavy lifting in that sentence. It's just simply the case that you're typically not going to have that amount of data available unless you're modeling a chemical with just an enormous database. So absent that, you can apply models that have a biological interpretation. So for dichotomous data, the dichotomous Hill model, if you're modeling a saturable process with Michaelis-Menten kinetics, if it's continuous data, you can use the Hill or exponential model for receptor-mediated responses. You may not know what the mode of action is for a chemical or an endpoint, and so you don't know which models have a biological interpretation. There may be a policy decision in place at your organization. So examples of this are the EPA's Integrated Risk Information System used the multi-stage model for cancer data because it provided consistency across cancer assessments, and it's a highly flexible family of models. EPA's Office of Pesticides program still uses the exponential model for acetylcholinesterase inhibition data, as far as I know. So these are policy decisions that drive modeling choices. Now, you may not have biological interpretation, you may not have policy decisions, and so otherwise, model fitting is in selection, defaults to a purely mathematical statistical process. The technical guidance lays out a approach for single model selection. The field has moved past this, and model averaging is where other software products have moved, and where BMDS is moving. We have model averaging for dichotomous endpoints. We are hoping to release new model averaging methods in the next version of BMDS for dichotomous and continuous. So BMDS is also moving towards this approach, and model averaging is identified in the technical guidance as an appropriate alternative approach. It allows for the synthesis of estimates from multiple models instead of picking one single model to the exclusion of all others. So again, an example of where the technical guidance sort of prognosticated where the field was moving. Lynn went over a number of approaches to improving model fit that the technical guidance touches on: removing the high dose, transforming doses, use of PBPK models to linearize saturable dose responses. For continuous data, the technical guidance does address using normal distributions versus log normal distributions for the continuous responses. So a number of miscellaneous topics that are covered, and that other issues or topics covered by the technical guidance that have actually been implemented in BMDS are the use of the Rao-Scott transformation for estimating and addressing litter effects. So in a situation where you're modeling a developmental endpoint, but you don't have the individual dam level data, you can use the Rao-Scott transformation to estimate the correct variance. Now, it's mentioned one time in one sentence in the technical guidance, but subsequent publications by EPA scientists elaborated on the transformation's use and provided a framework to apply it that has been implemented in BMDS. Modeling survival data, where you have differential survival across dose groups, which is common for cancer bioassays, is also addressed in the technical guidance. Again, no specific recommendations were provided in the guidance, but the approach has been applied in BMDS, and it has been used in risk assessments subsequently. So 2 examples where these sort of hints at approaches that are appropriate, where BMDS has subsequently taken up those approaches and implemented them. Cancer modeling. This is an example where a technical guidance document, a technical memo that was released by EPA's Statistical Working Group was released after the technical guidance and augmented recommendations made there. So this memo lays out an alternative single model selection approach for the multi-stage model when you're using it preferentially for cancer data, and just addresses the statistical issues that occur when you judge model fit when model parameters are estimated on a boundary. So just an example of some augmentation to the EPA's technical guidance outside of the actual technical guidance document. So conclusions, it's just impossible to not acknowledge that the technical guidance is almost 15 years old. It is rapidly, or it has already approached the limit of its useful lifespan. It's critical that new guidance is released on these now established methods that were only passingly referred to in the technical guidance. So EPA really does need guidance on Bayesian benchmark dose analysis, Bayesian model averaging, covariates, poly3, Rao-Scott. Needs to augment its incorporation of PBPK modeling explicitly within the dose response framework instead of treating them as 2 related quantitative approaches. And then, as probabilistic approaches for dose response modeling have been implemented, EPA needs guidance on the development and the use of probabilistic toxicity value derivation approaches. So technical guidance is still relevant, still useful, but it's time that new guidance comes out. So, and that is my presentation. Are we taking questions now or waiting until?
Yes, thanks so much, Alan. We are going to hold questions until the discussion at the end of this session. So as a reminder to everybody, please add your questions into the Q&A box and we'll ask them during our panel discussion. I would like to introduce our next speaker, Jose Cortinas Abrahantes from HESI, who will be talking about HESI 2022 BMD guidance, expectations, practical implementation, and harmonization. Welcome, Jose.
Thank you very much, Claire. Do you hear me well?
Yes, and we see your slides.
Okay, thank you very much. Yes, I'm going to talk about the HESI BMD guidance that was basically updated. The second update was made in 2022. First, also as well as Alan, I will just start with a disclaimer. This is just basically a presentation that is the sole responsibility of the authors of this presentation and do not reflect the official position of HESI. If you want to know the official position of HESI, you can go to the website of HESI. Thus, the outline of my presentation is as follows. I will start putting things a little bit into context, more or less repeating a bit what Alan was just presenting before, just introduce the methodological changes that were introduced in the guidance of 2022. Then, through examples, I will just provide some idea about what are the gains when we are shifting to the Bayesian modeling in the specific context of the BMD guidance of EFSA. And I will talk about a little bit about interpretation, what are the things that we would like to be reported whenever BMD analysis is performed, and I will end up with some conclusions. Thus, in terms of context, what is the purpose of our guidance? Well, it's just to try to provide a harmonized and transparent framework. Together with the guidance, there was also another technical document that was published in the EFSA journal with the full description of the methodological framework that is behind this guidance. And the intended audience of these guidances are applicants, risk assessors, scientific researchers, or consultants that are just basically dealing with these type of risk assessments. The guidance cover continuous and quantal endpoints. It introduces some idea about the biological plausibility of the dose response curve through something that are called soft constraint in the case of a Bayesian paradigm, and as well in relation to the relevance of the endpoint. And here we are referring to the selection of the BMR need to be just basically biologically based in the first step, but there is a tier approach that is presented in the guidance, and as well also in the selection for modeling of the endpoint. They need to be also relevant to the subject and the study that you are conducting. There has been also an introduction of a data suitability process, basically just assessing the design of the study that you want to analyze, just to understand the expectation in terms of how uncertain your estimation is going to be at the end of the day. It's only a flagging procedure, it's not basically a stopping rule. And then there we basically just propose an appropriate statistical methodology for dose response modeling. The evolution of this is that model averaging basically is introduced in this specific guidance, and which basically address very nicely uncertainty and provide as well something that most of the time we are using when we are using the frequencies approach, which is a probabilistic interpretation of the confidence interval, which is basically run in the context of a statistical analysis because they do not have a probabilistic interpretation. Thus, why BMD modeling matters? Well, basically because it uses the entire dose-response relationship and data, in contrast with the single-dose methods. It addresses uncertainty through the credible bounds that are basically produce the full posterior distribution of the parameter of interest, in this case, the BMD. It basically just, as we just provide an implementation of this specific framework, thus we try to ensure consistency across evaluation because they have a user-friendly interface that they are able to use to make this assessment. And we are also promoting transparent regulatory decision. Thus, we have as well just within this tool, we have provided also as well a default report of your analysis with all the information that we wanted to be able to evaluate whenever you submit a BMD evaluations to us, which make the assumptions visible. They are testable because they are also tests that are performed within the application. And then we are able to support any decision that are made from this analysis based on science-based regulatory decision. Thus, then what are the methodological changes that are introduced in 2022? Before going to that, I will give a little bit of history of the BMD in EFSA. We started a little bit later. USEPA started 2000. We started in 2005. There was not yet a BMD guidance, but we used it to obtain a margin of exposure at the time. In 2009, the first guidance was introduced, and there was based on single model selection and based on goodness of fit, the likelihood ratio test. And then in 2010, a workshop was basically taking place, and the two tools at the time that were PROS and BMDS were shown to the participants and how to use it in the context of BMD modeling. 7 years later, the first update of the guidance was produced, and there we were introducing already model averaging. It was already mentioned before by Alan as well that the usage of model averaging to account for model uncertainty, because we have several equations that we are able to use within this model, BMD modeling, and selecting one of them basically will just undermine the model uncertainty. Maybe all models are equally possible because we don't know what is the true underlying modeling that should be used. And 5 years later, following the update of the chapter 5 of dose response assessment in the EHC 240 from WHO, then we try to align with these specific recommendations in which they were recommending Bayesian model averaging. And in order to align with these recommendations, then we update the guidance and introduce Bayesian model averaging in this context. Thus, this is a little bit of a history of the BMD in EFSA. Just the structure of the guidance. The guidance provide just specific sections, just describing the framework, very succinct, because there is a technical report with a full description of all the framework that is basically just used in this guidance. And in the section 2.6, then we just provide some guidance to the user on how to use BMD and perform BMD analysis. And in the appendix, we have several examples illustrating how to use this methodology. We also complemented this with a flow chart with all the different steps that you should follow when you are conducting BMD analysis. Okay, I'm not going to go through all this, but that is also provided in the guidance. There's also some key concepts and definitions that have been already given before. I just want to make sure that the people understand that whenever we talk about BMD definitions and the BMRs used for continuous and quantum endpoint, in the case of continuous, we are basically estimating the median, not the mean. That sometimes is a misconception. There's the relative changes with respect to the median. In general, people are assuming log normality. That's why the median is the measure the centrality measure to use. In the case of quanta, well, the extra risk we have seen already from Alan why this is the measure to use. This is basically the two different definitions that EFSA uses are recommended to use. Okay, we just move away from one standard deviation because we consider that is also as well design dependent, depending on the study that you are going to be using to derive your BMD evaluation. Thus, if you use the standard deviation of the control that is in your experiment, it depends on the number of animals that you are using. It depends on the variability of that response that you might not be able to estimate it correctly based on the control data that you have in that specific experiment. That's why we just deviate in that cases from the US EPA guidance. We recommend to use then, in the specific case of continuous, the relative definition or the relative changes in respect to medians. Thus, then we use the BMDL and BMDU. The BMDL is the candidate reference point, and basically just if you always construct a 90% credible interval in this case, because we are using Bayesian paradigm. But what does it mean is that below this BMDL, there is still 5% chance, in this case I can talk about chance, that the BMD can be below that lower bound. And the BMDU basically is just saying that above this BMDU, there is still 5% chance the BMD can be above that specific threshold. Thus, these are the definitions which are having now a probabilistic interpretation in the case of Bayesian modeling. If you are doing it using frequentist approach, the interpretation of these lower and upper bounds cannot be giving a probabilistic interpretation. Okay, thus, the shift to Bayesian model averaging. Why? Because it basically reflects model uncertainty. It has as well the idea of not only model uncertainty in terms of equations, but also in terms of across distribution, because here we are also doing model averaging across different distribution. In the case of a continuous endpoint, we are considering normal and log normal distributions. This is also as well why we are shifting towards Bayesian model averaging. It's natural to consider that. It's just through the incorporation of priors and weighting schemes. It provides a transparent regulatory framework in which the prior distributions are basically described. In the technical document, there is also as well all the information in relation why we have chosen the priors for the specific parameters of those default priors. We have considered several prior distributions in that context, and then finally just evaluate through a simulation studies which one were the priors that we should use in this context. It's basically just you are able to construct posterior distribution for all the parameters of your models and the parameter of interest of the BMD, which just make it inference auditable. It improves interpretability. As I said before, now the BMD and the credible intervals have probabilistic interpretation, which is not the case in the Frequentis framework, because in the Frequentis framework, you should interpret the confidence intervals if you would repeat the experiment 100 times, and you would conduct these 100 analyses for each of the different experiments, in which the experiments are just supposed to be conducted under the same conditions. In 90 of those experiments and confidence interval, the true underlying BMD will be contained within that interval. But if you would just consider a single analysis, the BMD will be there, inside, or not. The probability is or 0 or 1, not 95%. Okay, this is the true interpretation of a Frequentis confidence interval, which is different from the Bayesian interpretation of the credible intervals. Thus, the method also allows to incorporate additional information through priors. Thus, you can also add, for instance, historical control data to estimate your background with high level of precision. Also, as well, other studies that were conducted, you could also incorporate priors for the BMD and as well then enhance the precision of that estimation. Thus, and in this specific context, then we are able just to introduce what we call a soft constraint. We have already heard about this several times of the constraint for the D parameter to ensure biological plausibility. Thus, here we are able just to introduce what we call a soft constraint through the priors. I will talk about that a little bit later with an example also. Thus, the workflow that we are basically just very briefly described, you saw how complex it is. It started by defining what type of data we are just going to be analyzing, favoring always to use individual data in case that is possible, because it allows us to check the assumptions that we are making. There are some underlying assumptions that we are making whenever we are using this modeling. And then if you have individual data, you will be able to assess distribution assumption, you will be able to assess constant variance or constant coefficient of variation in case that you are using log normal. You could also relax these assumptions. We don't have an implementation in which the variance depends on the dose, but this could be also as well some of the future developments that we could consider in EFSA. In terms of benchmark responses, for continuous endpoint, we propose to use a biologically relevant BMR. This is the first step in our selection of BMR. And then we propose a tier approach, as it was already mentioned yesterday by Lina. For Quantal, we, as US EPA, consider that 10% is basically just the recommended extra risk, but this can be modified as long as it's justified. We introduce a data suitability procedure that basically just assess the design of the study in terms of the size, in terms of the sample size, in terms of also as well dose spacing that is used in the experiment. These are allowing us to assess something in terms of what is the expectation of the uncertainty and the BMD estimation later on. Thus, we have developed this procedure to try to just provide a flagging procedure about your design, the design of the study that you are analyzing. And in this context also as well, we just provide a set of conditions that you need to use when you are reporting a Bayesian model averaging results, with the unique set of candidates model for the two type of endpoint, quantum and continuous. It's basically thus recommended to report model uncertainty with the different ways of the different models and the BMD and the BMDLs, BMDUs for each of the models, and as well as the model averaging, reporting the diagnostic test that we are basically just recommended to use to test the assumptions of the framework, because this could also have an impact on the results that we are basically collecting later on. Thus, the implementation of HESI for the Bayesian model averaging basically just recommend discrete priors, uniform discrete priors for the different models. We have a set of 8 models for the quantal data and a set of 16 models for the continuous endpoints. In the case of the continuous endpoint, we have 2 different distributions. The number of models are the same, 8 models for each of the 2 continuous distributions. And then we are basically recommending to use a priori, if we don't have any information about if one model is more likely to be reflecting the dose-response relationship than another one, we just assume that all of them are equally plausible. Also for the natural parameters, which are for us the background response, the maximum or minimum response, and the BMD, we propose to use a pair distribution. We consider several distributions for the prior distribution for these parameters. And this one for us was giving us good results in the simulation settings that we tested, and as well provide an intuitive prior specification, because they are based on minimum, maximum, and most likely value. Which is maybe easier to just try to ascertain for a specific parameter in the model. The computational methods that we are using in this specific framework, we try to balance between speed and robustness. Thus, we consider Laplace approximation to get speed, and then MCMC and bridge sampling just to basically just produce robust Bayesian estimation. And we recommend to use the bridge sampling for the submissions to our assessments of BMD analysis. Okay. In the case of model fit and diagnostic, we assess model fit using Bayes factor, which is basically just a method that is used within the Bayesian paradigm. Bayes factor in the sense of assessing if there is or not dose response effect. We compare the null model with the saturated model, and you use the Bayes factor to assess this comparison. Also as well, to ensure that there is at least one model in this suite of candidates, which are A models, that fit the data well. Thus, we compare the best fitting model with the saturated one, and we assess that through a Bayes factor as well. There is a rule of thumb just to assess this, like you are using with p-value 0.05. Here there are criteria that are used in this content, and in the technical report, we basically just describe this rule of thumb that are used. There are also as well a set of other tests that are also considered in terms of checking some of the other assumptions, like I mentioned before, constant variance, constant coefficient of variation, and the distributional assumptions in case of a continuous endpoint, if they are normal or log normal. And this is also as well used to propose a kind of sensitivity analysis to assess violation of these assumptions. Then the importance of this implementation is that now you are able to introduce informative priors in your analysis to improve the precision of your estimation in the cases that you are dealing with data that are not providing you enough information to estimate the parameter of interest. But these require justification and sensitivity analysis in order to assess how biased your results may be. Thus, in the guidance in 2022, we also provide some criteria to assess uncertainty in your estimation at the end of the process. Thus, if you have that all the models that are in the suite of models, the 8 candidate models are basically producing a poor fit, that will be a criteria that will tell you something about the uncertainty on your estimation from that specific data set. Another criteria is the ratio between BMD and BMD-L, that if it is larger than 20, then it's considered too uncertain. If the BMD is smaller than is 10 times lower than the lowest dose, then it's also considered as well a criteria to highlight uncertainty. Also, if the width of the credible interval is larger than 50, then this will also highlight uncertainty issues. Here you have basically just a link to the website. The user-friendly interface is very intuitive. I am not going to give you a demo of the application, but you can follow the manual, which is also as well there in that website, and they give you all the different steps that you need to follow to conduct a BMD analysis. Thus, the next part is just providing with some example what is the gain when you shift to Bayesian model averaging instead of frequentist. Thus, we have heard a lot about biological plausibility and some of the constraint issues that were mentioned before. In Europe before, we were not constraining the parameter D to be greater than 1 because then the inference process will be suffering. And this is due to the fact that if you take any of the models, these are just some representation, and you take the derivative, then you get that for these models, for instance, the derivative, then the term here that is dose to the power of D minus 1 is basically the one that is taking care that if the dose is 0, then your derivatives tend to infinity, meaning that you increase infinitely at dose 0. And this is what people basically just say that is biologically implausible. Okay, thus, and this is the main issue why the people just constrain the parameter D to be greater than 1, because then if this is larger than 1, it remains in the numerator, doesn't go to the denominator. That is just to illustrate with the mathematical equation why the people are basically just constraining this mode of D to be larger than 1. But in the Bayesian framework, you can just introduce what we call it soft constraint, basically to the prior. You just use informative priors. They are considered weakly informative priors, in which you give very little probability mass of the D value to take values below 1. And these are just the representation of the ones that are using US EPA for the quantal data for this specific parameter, and some of the models that they are using in the quantal data for the Bayesian modeling that they are using there. And they give around 5% chance to be below 1. We have also conduct simulation study comparing the results of these two different options. This can be also used as a sensitivity analysis. HESI is giving a little bit more chances to be smaller than 1, something around 20%. But basically, just by introducing this prior, you are just from the start saying that it is more likely to be above 1. Thus, you are trying to just comply with that biological plausibility. Another example is, this is in terms of constraint. Thus, we were talking before about the constraining the parameter D to be larger than 1. Thus, in the frequentist approach, there are also some ad hoc constraints that are as well used. This is just an example that was used in the BPA studies that were published and used by EFSA in the assessment of BPA. And we realized when we were just conducting the analysis that, and this is the things in red here, that the parameter, the minimum or fold change was basically set to 0.7692. And we found this a bit intriguing. Thus, all of them were having exactly the same parameter estimate, and then we realized that basically was hitting the boundary of that parameter estimate. There was an ad hoc constraint, and the ad hoc constraint was 1.3. And we realized that if we just relax this constraint, then we were getting a much better fit. This is the green rectangle here, producing the results without constraint, also using the frequentis model. And in that case, we are also able to see, if we look at the AIC values, that they are smaller than the ones that were obtained before when the constraint was used. We were not finding a reason why this constraint should be there. We were also as well considering simulation studies to see, using this study to set the simulation study based on the Hill model, of this Hill model, that was the best fitting model in this specific context. We just run 1,000 simulation studies in which we use the Bayesian methods and the frequentis method with the default assumptions that they are making. And we see that from these 1,000 simulation studies, 1,000 simulation data sets, based on this specific study, we realized that the BMDLs obtained with the frequencies were always lower than the BMDLs obtained with the Bayesian framework. The uncertainty of the credible interval or confidence interval were wider for the frequencies cases. The coverage, although of the two methods, were comparable, but if we look at the uncertainty criteria that were just provided before, in the HESI guidance, we see that only 3% of those 1,000 simulations were basically meeting the uncertainty conditions. The case of the Bayesian, 84% were meeting the uncertainty conditions. Here are some of the basically graphs of the different simulations with the width of the confidence interval for the Bayesian and the frequentis method, and as well the left side of the confidence or credible intervals. Another thing that we basically just gain by just shifting towards the Bayesian model averaging is that then we don't need to rely on bootstrapping to just construct model averaging confidence interval. For instance, in the case, this is also a study that was used in the PVD opinion of EFSA. In this specific case, sample sizes are very small. And you can see then that by using bootstrap, which is an asymptotic method, then we have problem then in the construction of the confidence interval for the model averaging. And we have seen as well that in this case, the model averaging confidence interval was basically very uncertain. The lower bound is very close to zero. In the case of the Bayesian model averaging, you can see that the lower bound is much larger than the one obtained using the frequentis approach. Also, when we try to increase the bootstrap, we see that we didn't improve accuracy in that case. Also, it's because of the low sample size. There is also a finite number of curves that you could just feed to your data because of the restricted number of animals within each of the different dose groups. Thus, the good thing of the Bayesian approach is that now you have a posterior distribution, which is basically using MCMC methods to approximate the distribution, and it has less problem with lower sample size. This is also an illustration of already Alan was mentioning this type of design of studies, but still we need to, sometimes this is the only study we have and we need to analyze the data. This is just a simulation study in which this was basically just the actual simulation. We had information about many different doses, and we were able to simulate the data and construct the curve, and the true BMD was around 0.02. And if the choices of the doses and the spacings are not correct, and the design is not well done, then you can end up with this kind of information. That's which is also as well given here in this table. Thus, as you can see from the first dose tested, the lowest active dose, already you were reaching the maximum. And sometimes you encounter this design, although if we just look at the data generating mechanism, the D value was 1.2. Here we don't have any idea about what is the way the curve should go from the control to the highest doses. Well, we conduct an analysis with the frequencies and Bayesian method. This is for this specific data set. We can see that as well with the frequencies method, we basically tend to estimate the values of D to be smaller than 1 in these specific conditions. Bayesian estimated values for D for all of the model very close to 1.2. The median value of this posterior distribution were very close to 1.2 for all the different models. And you can see in terms of model fits, the frequencies and the Bayesian models are basically producing comparable results in terms of model fits, if you look at the curves. But if we look at the model averaging estimates of the BMD, you can see that also the frequencies basically as well estimate the lower bound to be what it is, the lower bound in the estimation process. Okay, it basically hit that boundary and just keep it there, and it basically produced very large, very uncertainty, very uncertain confidence interval. It just didn't miss the true BMD with the upper bound. In the case of the Bayesian, you can see that it's much more, much less uncertain. If we look at the 1,000 simulations that we conducted for this specific design, we see that all the confidence interval produced by the frequencies method are basically just highly uncertain. None of the simulations were basically compliant with the thresholds that we were considering the HESI guidance. There were issues when estimating the D values. All of them in general are basically favoring values to be estimated to be below 1. In the case of a Bayesian approach, because of the prior distribution, which is putting a mass that is larger mass to values that are above 1, then it produces biologically plausible estimates. The interval uncertainty in the case of the Bayesian approach, basically just 99% of the 1,000 simulations were basically compliant with the threshold that we were considering in the guidance. Thus, what are the things that we consider in the case that we wanted to just get some submissions of BMD analysis to EFSA? Well, first of all, we would like to have information about the data. I mean, those, the sample size of each of the doses, mean and standard deviation, standard error, in the case of continuous or events, and sizes for quantum data. We prefer individual row data because this allows us to evaluate the design and also as well for reproducibility purposes. The BMR rationale should be provided, the biological relevance for continuous data, extra risk definition for quantal, and 10%. In general, if you deviate, you should just provide justification for this. The software version, including the settings that you are using to generate your analysis, prior sampling options. We just propose to use the default set of models in case you don't have any justification to just concentrate on a specific set of models. And we also as well encourage sensitivity checks, sensitivity analysis, especially with just if there are some violations, the assumptions. In the output, we want to see also as well the full credible interval, BMD, BMDL, BMDUs, model weights, diagnostics results, and fitted plots. And we also as well wanted to know some rationale in relation to the endpoint chosen for the reference point and not only mechanistically just considering the lowest BMDL of all the analysis. What are the common things that we see? There's that selective reporting. They don't show us everything. This is basically just discouraged because then there is a problem with the transparency of the things that you are just producing and sending us. Benchmark response choices are often lacking the rationale from the biological viewpoint. That is also as well something that we're trying to encourage now. Also, some overlook of the variance, basically just some assumptions that are made, like constant variance, constant coefficient of variation. These are also important and can impact the results of the analysis. When they don't consider clustering, this might lead to very precise risk estimates. And in the case of the BMD, we just basically see that most of the people try to use a mechanistic approach using the lowest BMDL from the endpoints that are analyzed, and we prefer to get the rationale about why they are using that specific endpoints to set the reference point. As a conclusion, then, I think that, in general, we have seen many things that are just basically comparable between the guidance of US EPA and EFSA. They are now introducing also as well Bayesian, which is also good to hear. It basically just this framework allows us to explicitly characterize the uncertainty. If we look at now that they are also as well converging to Bayesian and model averaging, this is going to be as well just enhancing consistency and harmonization across the globe. There's a transparency in terms of reporting the methodology and any choices that they have made in the process is important. Scientific justification of any of their choices, and then also as well using evidence-based decision instead of mechanistic lowest BMDL, that would also as well enhance harmonization. And as a key take-home messages, I would like to say that we would like to just defend benchmark those as being the best method to do this type of analysis, especially if we are dealing with Bayesian model averaging paradigm. I think that will be a way forward. The Bayesian model averaging just recognize uncertainty as an integral part of this process, and it's not a secondary condition. Just transparency and reproducibility. You need to produce transparent, justified, and reproducible analysis in order to be acceptable. This framework now provides a probabilistic interpretation of the resulting confidence credible intervals, and as well embedded the biological plausibility in their conceptual framework, and it allows as well the inclusion of additional information through prior. And this is my last slide. Thank you very much. Do you have any question?
Thank you, Jose, for that great talk. I would like to invite Alan and Jose, and then also Efizio Salazzo, also from EFSA, to join us for this discussion. And I'm introducing April Cleaver from HESI, who will be leading this discussion. As a reminder, if anyone has questions, please put them in the Q&A box. Thank you.
Okay, hi everyone. We have quite a few questions. We'll try our best to get through them all, but thank you to our audience for submitting so many. I'm going to start out with some sort of bigger picture questions for you all. What challenges do you see with companies who are submitting analyses to both agencies? How can companies address the differences potentially between the guidances, and what are some successful strategies that you may have seen?
You go for it, Alan?
Okay, well, I have a simple answer is I don't typically see analyses submitted by companies. So I don't work in pesticide registrations. So I'm usually either doing the modeling myself or providing technical assistance to agency scientists to, how to model the data and how to, construct and justify their dose response decisions. So from the standpoint of like a pesticide registration where the modeling has already been done and has to be used in a regulation, I don't really have that much experience.
Okay. But normally I am just basically in the size of developing the methods. And some other people are basically using, that's why we produce the user interface, the friendly user interface. But I have come across to differences between different methods, frequencies and Bayesian methods. In fact, in some of the gains that I was just presenting here, you could see the difference between the two different methods results. Okay. I think that the important part is just to understand why that difference are there, and just to try to find a logical and scientific explanation of the difference between the results. I understand that the choices are different, the methods are different, and these all bring uncertainty in terms of what method should I use, what options should I use. But we are trying to provide default assumptions and default considerations in this method that produce consistent results across different analyses. Okay. Most of the time, whenever we observe difference between results are because data are poor, okay, in data poor conditions. If data are rich enough, across different methods, we observe very, very, very similar results. That's, even with different choices of priors and different choices of model suites.
Yeah. I'll also say that there's an open technical support. There's a what we call e-ticket. I think we call it e-ticket still, but it's a platform where users can get in touch with me now. I'm the last man standing at EPA with BMDS since Jeff and Andy left. But so I say I help agency scientists with, modeling choices and interpreting, modeling results, but we field those results from the public as well. And, I think the general recommendation is, it's always better to get in touch with us earlier about modeling decisions or complicated modeling choices or interpreting results rather than, not in submitting something to like OPP for pesticide registration or OPPT. If like data, if confidential business information is an issue, I've had, external scientists reach out to me with anonymized data. Hey, I'm modeling chemical X and I'm seeing this. How do I handle with, this particular tricky modeling situation? And I've provided guidance, and that does provide sort of a firewall between me as an EPA scientist helping with what to me is a generalized anonymous modeling question and then a specific registration that might be submitted. So we're always happy to help with questions about, how to use the software, but also, hey, what do you think is the best approach for modeling this data? I've only got, an N of 3 in each dose, group. Is that enough to model the data? Or I've got this like sort of edge case modeling output. Is the modeling here sufficient to support, X, Y, and Z?
Great. Thank you so much. I think this kind of builds a little bit on what we were just talking about. In a regulatory framework, how likely do you see agreement between the applicant and the regulator on the prior and the BMR? There seems like there could be some expert judgment or subjectivity, which would have an impact on BMD and risk assessment.
Yeah, I think there's always going to be, a lot of people want to treat like dose response modeling as a science only and sort of remove a lot of the nuance. But there's always going to be some subjectivity. There's always going to be, and we want there to be discussion between the toxicologists and the modelers. We don't want it just to be purely a mathematical process. So yeah, there are going to be differences in, BMR selection. Say, hey, for this chemical, I think we've got this biological rationale. We should use it. I alluded previously to, what Jeff Giff used to say about, the correct answer is what you get through peer review. And it's just incumbent to recognize that it's not a black box process. You don't just put numbers in and get numbers out. You need to understand the basis and the rationale for the numbers and the modeling choices that you've made, and you need to be able to support those choices. And so, if I'm looking at an analysis and someone says, hey, I think a BMR of 5% is what's justified here, and they provide a sufficient rationale, sure, yeah, that makes sense to me. If someone just goes, I'm going to use a BMR of 5% because it's lower, that's going to be more of a problem. So, users of the method just need to be aware that you've got to always justify what you've done. It may be an easy justification of this is what the technical guidance says. It might be a more in-depth, nuanced justification of pulling in biological information, expert solicitation, whatever. But you always have to provide a justification for your modeling results. And if they're sufficient, more often than not, they'll probably be accepted.
Jose, I don't know if you wanted to add anything, or we could move on. We've had two.
No, we can move on. I was trying to type some answers to the questions that were in the chat.
Yeah, no problem. Two questions came up pretty quickly in the chat related to study design, so I'm going to pivot that way. This has to do with older chemical data sets, where some of the older studies may not meet some of the modern study designs. And there's a question about whether or not you can pool studies to get a better data set for benchmark dose modeling, or you should only do the modeling on a single study, even if the study isn't the best design for the modeling that you have.
Yeah. So right now, and I know there's another question in the chat about implementation of meta-analytic abilities in the software. And I mean, right now, like I always say, if users want features in BMDS, like get in touch with me. Like, I can use specific requests for methods development to sort of support development work. So, but I need to know what those are. So I'm always interested in what users want, new features in BMDS. There's no meta-analytic ability right now. In PROST and EFSA, there's sort of a quasi meta-analytic approach where you can use covariates. You can, combine male and female data, or species, data. You can aggregate data like that and generate, strata-specific BMDs and BMDLs. You can do that in categorical regression, which is a related software product that EPA puts out for ordinal data, which is just, the dichotomous data is just the most simple form of that. There are dose response analyses, EPA risk assessments where we're not using a strictly analytic or meta-analytic modeling approach, but before we have gotten to modeling, we've statistically determined that combining data is appropriate. I mean, one example I'm thinking of is, we had, I think, subacute data where there was only an N of 5 per dose group, and we had rat data, mouse data, male data, and female data. And we did this tiered, are male and female mice similar enough? Yes or no? Are male and female rats similar enough? Well, okay, they are. are rats and mice similar enough? And so we were able to aggregate this data up to a larger data set of, n equals 20 per dose group, and then model that. And that's, a larger data set, smaller confidence intervals, less uncertainty in that data. But that was sort of a data processing step rather than a modeling step. And we had to justify why that was appropriate to do in our risk assessment, and that was approved. So it's just an example of an outside of the box modeling approach or data processing approach that the technical guidance is silent on, but works when you have that when you're faced with that scenario.
Also, I want to say that, for instance, for cadmium, EFSA just produces as well an ad hoc procedure for meta-analytic BMD analysis. Okay, we don't have tools for that, but you are able to do these things outside of the tools that we provided. In terms of if we are just basically combining studies, you can use covariate analysis. It's not strictly a meta-analytic approach, because in general, meta-analytic approach, you are considering the studies as random effects. Yeah, and then you assess you assess that through random effect models. Yeah, study effect, which basically just consider parsimony. And in the context of the EFSA tool, you could consider the studies. In general, you don't have many studies. Also, when you are dealing with meta-analysis, you are dealing with many studies. Most of the time, you are not dealing with many studies when you are dealing with BMD analysis. Thus, considering that as fixed effects in your model are basically reasonable assumption. If you are having 2-3 studies, you could consider that as covariate in your model. Thus, there is a possibility to do that in the Bayesian tool, in the Bayesian model averaging tool of EFSA, to consider covariate analysis, where the covariate is the study. Thus, we have different study effect, and we can assess if the different parameters depend or not on the studies. And then provide also as well credible interval for the BMDs for the different studies, if they are different or not. And then you're able as well just to see the results of this analysis. But yes, covariate analysis will be one of the things that you would do in this context, especially if you are not dealing with many studies. If you are dealing with 10, 20 studies, then a covariate analysis would not be recommended. You would need a different methodological framework, which we don't have implemented and available to users.
Well, I think this sort of neatly comes into the question again that we had about animal use, where we have a global, or at least North America and Western Europe movement away from vertebrate testing, but we've heard several times now that we need sort of larger groups of animals to support some of the BMD analyses, and maybe meta-analysis techniques could reduce the number of animals overall if you have these data sets. Could you speak on the question? I'm going to read it verbatim. Why did EFSA and EPA move away from the NOAC to BMD? Would NOAC not always be more conservative? Both agencies are moving towards less vertebrate testing, yet it seems BMD needs more species. So meta-analytic techniques, I think, would help reduce the number of animals, but the question stands again, moving towards BMD, does that increase vertebrate testing?
But no, you can also use BMD for in vitro data as well. I mean, there is no need just to use in vivo experiments and BMD analysis. So we have also conducted BMD analysis on in vitro information and try to go to internal doses or internal concentrations in a specific target organs, and then they are used to conduct the BMD analysis there, and then to translate it back to using reverse dosimetry to get the external exposure. There is a possibility as well of doing these kind of things without just conducting in vivo experiments. Also, one of the things I want to say, NOAC doesn't always produce more conservative results. This is also important because that was one of the things that they were mentioning at that time in that question. It doesn't always produce more conservative results. And in fact, it basically lacks information about the uncertainty of that NOEC or NOAL. Yeah, there is no information about the uncertainty on this specific measurement. And this is also important that in BMD we are able to just assess the uncertainty in our estimations.
And in fact, as you have fewer animals per dose group or replicates per dose group, your NOAL is going to move up because you have less power to detect a change, whereas the BMDL is going to move in a more health protective direction because the confidence interval, the uncertainty in that data is larger, so the confidence interval is wider. And yeah, I mean, BMD methods are applied in NIHS's BMD Express for modeling transcriptomic data. So, there's a wealth of, examples of BMD modeling being applied to, in vivo and in vitro transcriptomic data.
Okay, we have, I think, time for one last question, and I do appreciate the activity in the Q&A. Thank you very much to our audience for being so active, and again, we'll try our best to answer you guys as we move through this webinar, potentially through written feedback. But I'm going to end with sort of the Bayesian versus Frequentis. The question is, it sounds like agencies across different regions and sectors are all moving towards the Bayesian averaging model and away from models currently recommended in guidances. Can panelists confirm this is a general trend and update to guidances in general could be expected in the future?
Well, so the easy answer is yes, we are moving to Bayesian approaches in BMDS, and we've got methods that are currently under external peer review right now with the hope that we can implement these methods in the next version of BMDS. So, if this peer review goes well, users can expect to see new Bayesian model averaging approaches in BMDS. The question of guidance is a bit out of my hands. It would be great, and I would love for capital G guidance to be promulgated by EPA. A new document like the technical guidance. I don't know if that will happen, but I think in the absence of that, what you will see at the agency is a body of application build up across assessments, across program offices, where Bayesian model averaging and other probabilistic approaches are used in individual assessments that themselves undergo peer review. And as that happens, sort of a body of capital G guidance will develop. Lowercase g, I meant to say, sorry.
And you, Jose?
We already moved. We are already there. And in 2022, WHO updated the chapter 5 of dose response, and they recommend Bayesian model averaging. And from that moment onwards, we started to develop this framework and to produce a guidance. And in 2022, we already produced I think that the update was in 2020, not in 2022, sorry, 2020. And in 2022, we had then the tool and the guidance based on this specific framework. And now we are just basically improving it along with the usage, okay? And just incorporating new features and trying to enhance the reporting and these kind of things. It's an endeavor that it never finishes.
Yeah, I think as well, I'm curious that you are observing a trend in this direction as well in other of the international agencies as well. Like this is a general trend.
Yeah.
EFSA has already made it, yes. Okay. All right, well, we are at time and we're trying to stay on schedule. So I want to thank you, Alan and Jose, for great presentations and a very active Q&A session. So thanks again, and I'm going to turn it over to Claire.
Yes, thank you, April, Alan, and Jose. We are now at our scheduled break, so we are going to reconvene at 9:45 Eastern Time in about 15 minutes. When the slides get pulled back up, we will have a QR code shown during the break that if you're interested in filling out our introductory poll, just so that we know who is in the room and what your knowledge on BMD is, you're welcome to scan this QR code, and then we will be back in 15 minutes, 15 minutes from the top of the hour. See you all soon. Again, so apologies. So we are going to get started with our case examples and practical applications session. Our first speaker will be Matt Wheeler from Talk Strategies, who will be talking about addressing uncertainty in agrochemical assessment using Bayesian techniques for BMD and point of departure. So welcome, Matt.
Thank you. This is going to dovetail nicely into what Jose and Alan did, because it's a kind of a comparison between methods from HESI and kind of some of the behind the scenes stuff that's going on at the EPA. So next slide. I want to focus the fact that model uncertainty exists in a wide range of contexts, and it's going to impact the ultimate benchmark dose and point of departure. And though I'm going to do this in the agrochem and on the US side, OPP still has a suggestion of the NOAL. I'm going to kind of ignore that suggestion and just do it strictly in the idea of a benchmark dose. And then in terms of the uncertainty, I want to focus on Bayesian model averaging. And then, given kind of some of the things that were said yesterday about using epi data for dose response, I want to focus on something that's a little less thought about and the uncertainty there. So kind of prime the pumps for that type of idea. So essentially explore different ways to deal with this uncertainty in model averaging specifically, but like something that we're quantitative bias assessment and talking about that uncertainty in terms of the dose response and what it can actually do. So next slide. So this is our first case study. Next slide. What I'm going to do is take data that's directly available to all. So this is a data set that's, you can see in the lower right corner, that's essentially looking at very different biomarkers of animals, rats, 5 rats for all dose groups, exposed to pendimethalin. I think I pronounced it correct. And I'm going to compare what happens when you do model averaging, what happens when you look at the best model, and then EFSA's tools and Toxic R. So Toxic R is related to the EPA tool. It's actually got the same underlying code right now, but it's going to be able to do a little bit more. So next slide. So here's the analysis for alt. At the top left corner, this is actually what PROAST was reported in the study. And I want you to focus on the fact those two circles are cases where the given models, one is the exponential 5 and the other is the hill, don't really fit the data very well. They're actually missing the dose response curve. Now, when you start to use model averaging, you get a large suite now of models. Now, the truth is Toxic R is using a total of 15 models. I think it's the exact same suite used in EFSA. So it's a little bit different than what the EPA would use. And so if you look at the EFSA model, and it's kind of hard given the graphs, you hit the data, you describe the data much better. And that's because there's more models using the model average, and you get a very tight estimate of the BMR. Now I'm using both the SD approach and the default EFSA approach in terms of relative effect. And you're going to notice that they're going to be about the same answer. And that often happens in these cases. So next slide. So if you use EFSA, you're going to get a lower BMDL on 95% of 105.1. Toxic R is going to be a little higher, but that again is solely based upon the fact that you're using a different definition of the benchmark dose, 127.8 in terms of mg per kg per day. And then PROAST, the BMDL, the model range. So there was a bunch of different BMDLs given between 82.6 and 98.1. So it's actually going to be lower than what you get using the model averaging with the better model fits. So there was some question about, what happens, when you're dealing with regulatory things between Europe and the United States or other agencies. And the reality is when you start to use model averaging, things start to actually harmonize by themselves and you get answers that are qualitatively very similar. So next slide. And here's another case, SOD. And the reason why I picked the previous one was there was no NOAL in the previous one. Here's a case where there is a NOAL. So, and then the question came up, the NOAC, no observed effect concentration. In this case, you're going to see a little bit of difference because you're fitting the data well. PROAST in this case fit the data well as well. But again, your benchmark dose lower bound, which is what everybody's going to use, you can see that the lower bound on both cases is visually going to be just above 100. And we can go down to the next slide. And you'll get, in terms of this inflammation, you're going to get BMDs in the same range, almost the exact same thing. And here's the interesting thing. Now PROAST is giving kind of the opposite answer. It's putting it at 149, 151. So actually, in terms of agreement, model averaging is starting to have more agreement versus disagreement in terms of the actual estimate that you're getting. So these things are nice in the sense that things are starting to align in terms of the methodologies. And you'll notice also that realistically, for this example, most everything gets really close to 100, which is your lowest dose in the dose response. So there's going to be a lot of agreement between the no observed effect concentration and the benchmark dose in most cases. That's what's been reported throughout time, the past 25 years in the literature. The nice thing here is you start to become more agnostic in terms of the actual methodology used when you start to use a Bayesian model framework. So next slide. So PROAST doesn't give much more model uncertainty. The more advanced model averaging approaches give different estimates of the BMD and the point of departure. Now, I would notice if you did in the one case use a low AL, it's going to require an extra uncertainty factor of 10, at least for according to EPA guidance. So the 100 low AL now turns into a 10 meg per kg per day. So you're adding uncertainty in the idea that you had a low AL in that case, whereas you would stay in the 100 meg per kg per day range using the benchmark dose. Now, the use of model averaging using toxic R, the EFSA tool, is going to reduce uncertainty by accounting for it in terms of actually trying to understand what models are the best fit to the data. And it gets us around this idea. I mean, you saw even with the PROAST estimates that were given, there was as much uncertainty, if not more, in just the Hill and the exponential model than there was between the EFSA approach and the toxic R approach, even though they had different BMD, BMR definitions. So that's something interesting. And I think really the default BMR definitions of toxic R, of EFSA and the EPA, they both pretty much were developed to get about the same answer. They were done on different logic, but in the end of the day, they always usually give about the same answer. So I'm going to kind of switch gears and make a little bit of thought of like uncertainty in different directions. So next slide. So this is going to be very brief, very brief. Just here's the idea. What one of the big problems when we do observational studies, which was talked yesterday, is like we have a dose response of a chemical. So here's a chemical that I found, lindane, in the literature. And the question is, we have studies that often do case control studies, observational studies, where we study the chemical or the exposure very well, often using splines, using other things. But then we study these confounders and we put very simple things into the model. And over here you can see, for example, this is a lymphoma data set that I have in the right, but over here in the left, you see a very complicated relationship when BMI is actually studied. However, over on the right, what you have is a very simple relationship with usually BMI greater than or equal to something. You might get a couple groups. And that, if there's any relationship between the exposure and the other, you're going to have some issues in terms of your model fit, because what's going to happen is your exposure is actually going to pick up the difference. It's going to, because it's correlated between BMI, you're going to actually have a biased benchmark dose. So this is something that has to be thought about. And I don't know if it's always very well thought about. So I kind of tried to simplify it in the next slide to get some thoughts about it. Next slide. If you have such a thing between chemical exposure and BMI, and say this is just a simple case control study, and you have odds ratios here, you can actually start to look at the literature, make adjustments based upon these. These are Bayesian adjustments. And you can see now, in this case, this is a bias towards away from the null, the way this is done. But in the other case, it could be a bias towards the null, against the null, or towards the null in the result, because what could be happening is if BMI is incorrectly described, then you're going to average everything out, and the dose response is actually going to be less steep. So, and I've seen changes in significance. I've seen values go from 1e to the -6 to not significant 0.2 when you are just talking about slight changes. So next slide. I don't know if I have much time to go into it. So uncertainty in benchmark dose estimation arises in a variety of contexts. You can do model averaging to produce pointed departures, BMDs and BMDLs. Uncertainty in dose response is not limited to what we would call classical dose response data. It's very much evident in epidemiological studies. And as we move away from animal studies, these epi studies are becoming more important in the dose response. So we have to really think about how to use them and how to think about how these biases play a part. Uncertainty can be quantified, model averaging, quantitative bias assessment, all of these things allow us to quantify uncertainty, get better estimates of the benchmark dose in the end, but it's important to pick the right tool for the research question and to get the right answer for the regulatory context. So I'm thinking about time, so thank you.
Thank you, Matt, for that great talk. And as a reminder to everybody, please drop your questions in the Q&A box. We're going to hold questions until the end of the session when we have our panel discussion. And also for questions that were not answered in the last session, we're going to do our best to have our speakers answer them in the Q&A box, but then also can do some follow-up after the fact. So with that, I would like to introduce our next speaker, Manousos Foudourakis from Corteva, who will be speaking about using BMD for crop protection and industry perspective. So welcome, Manousos.
Thank you. Can you see my desktop?
Yes, we see your screen.
Yeah. Good morning, good evening, everyone. Today, Corteva AgriSciences and Syngenta are going to discuss the crop protection industry perspective on a significant shift on how we approach the ecotoxicological risk assessment for birds and mammals, specifically focusing here on the transition from the NOAEL approach to the BMDs. So actually is the next step how we can introduce the new maths in the regulatory environment. To set the stage, we must look at the regulatory and scientific context, the key data types and the study types that are used. In the avian assessment, we take the hazard points from the laboratory studies, we apply them to the exposure models, and these allow us to extrapolate from the control environments to the complex reality of the wild bird populations. For mammals, the concept is the same. We often use the same mammalian datasets that are used from the human health safety to reach the conclusions now regarding the wild mammalian species. The momentum behind the BMD adoption is largely driven by the EFSA. The concept was first updated in 2017 guidance, then with the 2022 guidance placed a greatest emphasis on the Bayesian approaches, and at the same time we have the BMD guide, birds and mammals guidance, which is the BMDs is cross-reference in this guidance for pesticides. So the BMD is formally integrated into this regulatory context as the default approach. Under the new guidance, EFSA considers it appropriate to use the benchmark dose of 10%, BMR of 10% for both quantal and continuous data. And this replaces the NOAEL for the long-term risk assessments. The BMD lower limit can be used also when there is an additional uncertainty. But we must note here that the 10% BMR is often has limited biological relevance for many of the parameters that we evaluate in these studies. The scale of this change is immense. We are now looking at all the in vivo dietary studies from the mammalian toxicology. When you have hundreds of potential BMD candidates derived from multiple parameters, the analysis becomes incredibly complex. The mathematical optimization introduces a substantial burden which requires complex evaluation and the involvement of numerous experts across stakeholders organizations to continuously interpret, clarify, and resolve the evaluation and the statistical issues. From the human health experience on pesticides in Europe, the BMD remains limited use and on a case by case. In North America, the US EPA accepts the BMD for in-depth assessments, though it isn't yet routine for all the pesticides in both US and Canada. Historically, pesticide risk assessment for potential carcinogens of unknown mode of action have utilized the care-fitting maximum likelihood estimates to extrapolate from tumor incident dose response data and generate cancer slope factors. In Brazil is also opening the door. The Anvisa recently released 4 tox evaluation guidelines, and the BMD is mentioned as a possibility, not as a requirement. Anvisa mentioned as a possibility provided that it follows the EPA and the EFSA guidelines. The transition brings 4 major challenges in the regulatory. It's the study design limitations that we have heard also before, the large volume of data versus the meaningful endpoints in ecotoxicology, technical challenges for the tools, and the regulatory and procedural hurdles. The first hurdle is the study design. So yes, reliable BMD calculations, as we have heard, require a high number of test levels and a clear dose response. If these conditions are satisfied, then yes, the BMD calculations can be reliable and the BMD10 can replace the NOAEL. The more legacy avian and mammalian chronic studies were not designed this way. They often lack these sufficient dose levels, meaning that in many cases, a reliable BMD then cannot be calculated and we must default back to the NOAEL. So it is highly unlikely that the BMD10 will ever replace completely the NOAEL values in the regulatory environment because of the study design challenges. Then there is the resource burden. From our initial estimations, we have seen typical assessments take anywhere from 150 up to 1800 hours, averaging over 600 hours per substance to complete a BMD analysis. And this creates an enormous demand for both applicants and authorities, often for points that really lack ecological relevance. So increased resources are needed for all the stakeholders. So how shall we handle a large volume of data? So this is bring us to the distinction how we can filter the data between apical and the interim effects. For example, here in a multi-generation study, the maternal body weight is measured frequently every 7 days during gestation and lactation. And transient weight reduction, let's say at the 14 days of gestation, is likely relevant to the population, which is the protection goal here in ecotoxicology. We should focus on the apical endpoints, such as the weight at the end of the lactation, which actually impacts the reproductive output and consider the most ecological relevant. Modeling every interim measurement adds works without adding any scientific value. The software stability also is a major concern. The EFSA Bayesian tool on the R4EU platform is frequently updated. However, the absence of a stable regulatory version, proper version control and transparency causes major problems. In addition, we continue to experience bugs, real-world datasets which cannot be analyzed, and suspicious results. We have seen, not now, in previous tool version, an XBMD value in one version and an almost 2x value if you run the same dataset in the following BMD version of the tool, which is completely unacceptable in the regulatory. So robust validation using a variety of real-world data here is required. Without a stable regulatory version, as we call it, an analysis done today might yield a different result tomorrow, and this lack of transparency and version control makes it really difficult to rely on the tool for those HESI submissions. We are currently working with EFSA to improve this, but the problem is not yet fixed, and the platform is not fit for purpose for reliable implementation in the regulatory. So it's critical to have a thoroughly tested regulatory version of the tools which works reliably and consistently if we want to advance them into the regulatory. Furthermore, we are moving to the Bayesian statistics, which adds a layer of a black box complexity. Most ecotoxicologists are not Bayesian statisticians. The choice of priors is subjective, and the complexity outweighs the benefits for many standard assessments. Any benefits brought about by the use of Bayesian are outweighed by the drawbacks. What we really need is a globally harmonized approach. If the BMD is the future, we need a single, robust analysis that can be accepted in Europe, in US, and beyond, such like we do with ECX values today. We should work to ensure that a single, robust analysis can be consistently accepted across all the regulatory jurisdictions. The current landscape is defined by transition pressure. We face inefficiencies as inconsistent practices by different evaluators lead to redundant work and submission inefficiencies. We face also a need for higher expert knowledge. Increasingly, the BMD tools are used by people who may not have this deep technical knowledge, and a scientific robust evaluation might not always be possible due to this lack of expertise. The BMD is implemented since October of 2025 in Europe for birds and mammals. The applicants face inconsistent practices across the member states, so it's really challenging to adapt planning, modeling workflows, and data interpretation. So increased expertise needed for all the stakeholders and harmonized approach. Let's look the practical impact on a project timeline. I will present how the BMD fits now in the regulatory field. The process of generating birds and mammals data for an EU submission involves specific regulatory steps. Ideally, the preparation should begin 3 years before the regulatory submission to ensure that you have a comprehensive approach. And during this time, the first priority, of course, is to complete all these GLP studies across mammalian toxicology and avian ecotoxicology, which means that you have to start generating the data 2-3 years before. Following the finalizing also the proposed label, which is a critical step, not only for birds and mammals, not only for ecotox, but for the entire dossier. The process begins with an in-depth review now of all the studies. You run the BMD calculations for each of these endpoints. You pick up the one that you think that it is relevant for the risk assessment. You conduct the default risk assessment. And if you fail, either you will adjust the label or you will proceed in planning higher tier studies. There are field testing in order to address all these outstanding risk factors. So starting early and following all these steps, it ensures that you will have a well-prepared submission and helps to avoid any delays or setbacks in the regulatory review process. At the 2-year mark, if this regulatory process it's crucial to execute this first package of higher tier field studies. We call them focal species studies, where we observe the species in the field, assuming, of course, that there are no changes in the BMD tool. As an example that we have seen before, if the BM tool has been updated, then we need to reconsider everything. Our field testing might be invalidated, and in this case, we need to rerun the BMD calculations to confirm that the values remain stable. And of course, the biggest challenge arises if the values have shifted. And if that happens, it becomes necessary to return to step 3 and 4 from the 3 years mark, redo the risk assessment, check if you fail in more species, readjust your plannings, or plan for more field studies in order to address the additional risk. At the 1-year mark in this regulatory process, we move forward to the second package of field studies. We call them ecological data, assuming, of course, that the BMD again remains unchanged. It's the first time now that you will meet the authorities, and it's the first time after many years that you will discuss the authorities the BMD results in what we called pre-submission meetings with the rapporteur member state. However, of course, if the tool has been updated, we may face new challenges, and in some cases, we may have to start and rerun the BMD calculations to confirm the values, which may bring, of course, a cascade of new challenges and potential forces to return to the early stages of the planning. This is why we noted before that the BMD estimation is a pillar in the development of the strategies for all these years of higher tier studies, and why completing the label years in advance is so essential. This is why that a stable tool is not just a technical preference, it is a business necessity. In the final submission year, our focus shifts to compiling all the information, write and submit the dossier, and then they will have the follow-up discussions with the rapporteur member state, the EFSA, in order to conduct evaluation and all these technical meetings. So a lack of alignment for so many years process, 5-6 years that you have seen on the BMD evaluation and tools, which will require to recalculate the BM tools and potentially setting off a chain of issues that could disrupt and jeopardize the entire process. In summary, the regulatory challenges are significant. BMD is the foundation of the risk assessment. It needs to help design and execute all these higher tier strategies and finalize the label. Yet the predictability of all these things is severely impacted. Even after submission, probably you need to revise the strategies again And all the delayed results hinder the applicant's ability here to generate this data and adapt on the process in a timely manner. The risk assessment outcomes becomes unpredictable. It affects the whole process. And to conclude, we must prioritize the ecological significant over the interim effects. In the EU, we're shifting to the BMD 10, but we must recognize that many studies cannot support it, and this tradition requires more resources, more expertise, and above all, a harmonized approach to ensure that our risk assessment remains predictable and scientifically sound. Thanks a lot.
Thank you, Manousos. We appreciate the talk. Again, please drop your questions in the Q&A box. And I would like to introduce our next speaker, who is Paul White from Health Canada, who will be speaking on research investigating the utility of the BMD approach for chemical safety assessment. So welcome, Paul.
So thanks for the invitation to participate in this workshop. I'm going to talk to you this morning about research investigating the utility of the benchmark dose approach for chemical safety assessment.
Thanks, Paul. I just interrupt really quickly. Apologies. Would you be able to turn your camera on so that people can see you while you speak?
Oh, I'm sorry. I didn't even realize that it wasn't on. How's that?
Great. Perfect. Appreciate it. Thank you.
Thanks. So, and as mentioned, I'm from Health Canada in Ottawa. I'm in the part of Health Canada that's called the Environmental Health Science and Research Bureau. So I'd just like to start off with a disclaimer. So I wanted to emphasize that the views expressed in my presentation are the results of ongoing scientific research, and the views in the discussed research results don't necessarily reflect the current or anticipated policies or recommendations of Health Canada. So many people have mentioned the late Wout Slub. I wanted to mention him as well. Wout and I worked very closely together. He'll be sorely missed. I talked to him only a few days before he tragically passed away. We had been working on some BMD analysis of mixtures data. So yeah, Wout and I were pretty close friends. Just jumping right into it, some of the work that I've been doing is to look at model the performance of different models from the BMD analysis point of view. So several people have talked about study design and sorry, not model, but study design. So we have been looking at different study designs and the influence of study design on the BMD value. So we've been working with in vitro and in vivo data, some work that we've been doing with Lightron laboratories. The in vivo data was data that was generated by Alexandra Long in my lab. The in vivo data, what I'm showing here on the left is the details of what we're calling the master data set or the full data set. It was a large study on benzoapyrene, a 28-day repeat dose gavage study in mutamouse. And we looked at 11 doses, so 10 doses plus control with 7 animals per dose. So that was a very large study. We have sort of an equivalently large in vitro study on temozolomide with 14 concentrations and 6 replicates per concentration. I'm not going to talk about the analyses of those data that looked at a number of different genotoxicity biomarkers. So what I'm going to talk to you about is what we've done with the in vivo data. So what we did was repeated sampling from what I'm calling the full data set to create new study designs with simulated data sets. So this is showing you a dose response for bone marrow for the full data set with LAC-Z mutant frequency against dose. And you can see here's the dose response plot with for the 10 doses plus control with the BMD value. And then what we did was we used the full in vitro or in vivo study data sets and repeatedly sampled those data sets to simulate smaller study designs. So dose spacing, and replicates per dose were varied iteratively to construct new study data sets and then use the benchmark dose approach to fit the function to the data, look at the benchmark dose and compare it to what we had observed for the full data set and see whether it's within the confidence limits of the benchmark dose for the full data set. So the BMD analysis was used to evaluate study designs relative to the full study design or the full data set. And the full is an 11 by 7 study design. And here you can see it So it's an 11x7 study design, and you can see two examples. I mean, we did this 1,000 times for the data set. There's a 4x6 study design and an 8x3 study design, 4 being the number of dose groups, 6 being the number of animals per dose. And so we did that iteratively, and I don't have time to talk about the details. I'm just going to give you the key messages of those analyses. Overall, we found that traditional study designs with few dose groups and more animals per dose group had relatively poor precision. So the simulated BMDs were often outside the 90% confidence interval of the full study BMD. And I think this is well aligned with what some other people have been saying in previous presentations, both today and yesterday. Studies with the same number of experimental animals or experimental samples distributed across more dose groups generally generated more precise BMDs. So we can compare the 4x6 design, which is fairly common, to a 6x4 design, so 6 dose groups, or even a 7x3, where the 7x3 would actually include fewer animals. And so it gets to this 3 R's principle, and we get a more precise BMD with fewer animals. So the results imply that the distribution of replicate animals across more appropriately spaced dose groups is a better use of experimental animals since it maximizes BMD precision. I think where things get complicated is this sort of tug of war between the study designs that are recommended in the test guidelines and study designs that give you a more precise BMD. So for an in vivo study, if a 4x5 or 6 study design is a necessity because you're following guidelines like test guideline 488, then dose selection and spacing is obviously critical for obtaining a precise BMD. But in large part, these analyses resulted in basically saying that you're better off distributing your animals to more dose groups than more animals per dose group from a BMD precision point of view. We've been doing a lot of work also on determination of benchmark response values. I think many people are familiar with Weltzlaub's general or effect size theory, where he basically describes a method whereby you can calculate a benchmark response that's meaningful for a particular endpoint. And what I'm going to talk to you about is some work that I did with Guangqiao Chen, who's going to be one of the speakers coming up this morning. Here's an example dose response plot of reticulocyte concentration against dose. And what's important here is what it's showing is the maximum response and the within group standard deviation. So those, Wout would argue, are unique properties of a particular endpoint, and you can use the values that are determined using the benchmark dose approach to determine or calculate what would be a meaningful benchmark response. And so here's, I won't go into the details, but here's the equation that you can use to determine a benchmark response based on within group standard deviation. That's S there in that equation. And what we've done here, and we being Guangqiao and I and a series of a bunch of other people, we've used dose response data from a large number of studies to determine what S is or what M is, and then use the equation to calculate a benchmark response for that particular endpoint. So one of the things that we did was we wanted to see if S or M was stable for a particular endpoint. So one of the things we did was we looked at the influence of experimental factors or covariates on S or M, like administration route, tissue, treatment duration, and post-exposure tissue sampling time. My computer's going a bit slowly here, on M or S to see if S is stable for a given endpoint. So this, very quickly, we did these analyses for a couple of different endpoints, one being the transgenic rodent mutagenicity assay and the in vivo pig A mutagenicity assay. We analyzed 171 data sets for TGR and 172 pig A data sets. You can see the results of the benchmark dose analysis. The plot on the left is for TGR. You can see mutant frequency against dose for the 171 dose response analyses, and then on the right for the pig A dose response analyses. And we use data set number as a covariate for potency and background. And what we did was we estimated a single var value or a single S within group standard deviation value with which to calculate the BMR for that particular endpoint, either TGR or pig A. And when we did that very quickly, we determined, well, I should mention here that we actually used two methods. So we used a method for BMR determination based on VAR, the within group variance. but we also used what I guess you could call a variation on the 1SD approach, using what was described in Andreas Zeller's 2017 paper, where you can look at historical controls or study-specific standard deviations to also get a handle on what would be a rational benchmark response for a particular endpoint. And this table is just very quickly showing you an overview of what we found. So here's the VAR value. Here are the BMRs based on S, 47% and 60% for those two endpoints, and the BMR values based on study-specific background standard deviation, so standard deviation of the controls. And you can see that the values deviate substantially from what some others have used, even in the genotoxicity literature, 5% or 10%. So what we stated in the manuscript that we published last year in 2025 is that what makes sense, in our opinion, for mutagenicity endpoints in vivo is 50%. And interestingly, 50% was already being used in the genotoxicity literature, and I've shown here, I think there are 9 papers that predated our 2025 paper. And I think it's probably because we presented this work earlier at the HESI Genetic Toxicology Technical Committee meeting, and people started using it even before we published the results of our analyses. And I think what we need to do is continue these kinds of analyses to determine what would be effective benchmark responses for other genotoxicity endpoints, like for the COMET assay for DNA strand breaks, or for the mutagenicity assay based on error-corrected next-generation sequencing. So with respect to ECNGS, we've actually done that. So we've done the analyses analogous to what we did for TGR and PIG-A to determine a BMR value for that particular in vivo mutagenicity endpoint. And we've looked at data for duplex sequencing, HOC-seq, PEC-seq, and PacBio HiFi. And here you can see the plot for the duplex sequencing data analysis, mutation frequency against dose for 47 data sets. So this work now is in press. The first author is Sarin Mulughetto. And what we found using the equation that I showed on the previous slide with the 47 duplex sequencing data sets is we get a very precise VAR estimate, and we can use that to determine the BMR based on S, which comes out to 27.7%. If we look at, and I should mention that we also found no appreciable effect of the covariates on VAR. So all these covariates that are listed here, we found that VAR was very stable for that specific endpoint. When we look at the benchmark response based on the standard deviation of historical controls, we get a value of 28.8%. So these are very consistent BMRs using these two different determination approaches, and they're lower than what we had specified before, but not by a huge amount. So we're talking here 30% range instead of 50% for the other in vivo mutagenicity endpoints. So skipping right ahead, one of the other things that we've been doing a lot is we've been using the BMD covariate approach to look at compound potency ranking. And here I'm showing you the results of model averaging for 4 nitrosamines. This is big blue rat liver data. So it's C2 mutant frequency against dose. And you can see the results here for 4 different compounds: nitrosapiperidine in green, nitrosamine or nicotine-derived nitrosamine ketone in red, nitrosadeloxetine, and NTTP. So you can see this marked transition or marked change in BMD values, with nitrosapiperidine being the most potent with a low BMD value all the way to NTTP. And I'm talking about this because it's important to think about how you could use these relative potency values. So this potency differences in compounds. What people are proposing now is that you can use relative mutagenic potency to adjust carcinogenicity-derived acceptable intake values for N-nitrosamines that are noteworthy impurities in pharmaceutical products. So here I'm showing you data for nitrosaroboxetine and nitrosamorpholine, nitrosamorpholine being the much more potent in black. And you can see these marked differences in potency shown on this diagram for nitrosamorpholine and nitrosaroboxetine. And what some people are proposing is that you could use this difference in potency to determine an acceptable intake for a compound that hasn't been tested for carcinogenic activity. And you use what people have been calling an anchor molecule or an anchor compound to calculate, and I guess you could call it an estimated AI. And so very quickly, I'm not going to go into it any detail, people have done this. I got the data here from Xiaofei Zhang at Pfizer, and they've done this for this particular example with nitrosomorpholine and nitrosoroboxetine, and calculated an AI of 24,000 instead of, if you use read across, 129. But it's really important to note that although this approach is being presented in the scientific literature, I was able to find 5 papers where people are doing it. It's not currently accepted by regulatory authorities, but there's a lot of discussion. So it's an interesting way of looking at how you could determine acceptable intakes for compounds that haven't been tested for carcinogenic activity. And people are scrutinizing the utility of the approach, and there's a lot of discussion. So aligned with that discussion is the broader relationship between carcinogenic potency and mutagenic potency. And George Johnson showed a variation on this. I'm not sure if he showed this exact plot showing the relationship between cancer BMD and, in this case, mutagenic BMD or genotoxicity BMD measured as micronucleus inducing potency. And you can see this strong empirical relationship. So the question that remains is, does this empirical relationship exist for other endpoints? So, for example, the transgenic rodent gene mutation assay or the error-corrected sequencing, the error-corrected next-generation sequencing mutagenicity assays. So with this, very quickly, with this particular example, they found that there was this relationship between lowest carcinogenic potency, BMD, against lowest genotoxic potency, and that for 21 of the 23 compounds, there was a lower BMD for micronucleus-inducing potency than for tumor-inducing potency. So higher potency for the micronucleus endpoint. And there were 2 compounds that showed the opposite trend. So what's important here is that in this paper by Leah Hernandez, they're proposing that you could use genotoxicity data to predict carcinogenic potency with an uncertainty factor spanning about a factor of 100. So we've been looking at the relationship between carcinogenic potency and mutagenic potency measured using the transgenic rodent gene mutation assay. And we've been using data that are in a database that we've built called the transgenic rodent dose response database. And you can see that it includes a really huge number of observations across a large number of data sets. So it gives us the ability to look at this question about the relationship between CARC potency and mutagenic potency very rigorously. So we've been doing this actively now. I should mention that there's a lot of variability across these different experimental factors, so it complicates the data analysis. We've looked at the carcinogenic potency database, and we were able to find 78 compounds that have both TGR data and carcinogenic potency data, and we're currently doing the analysis to look at the relationship between these two. I can't show you the results because we don't have them yet. There are 13 predominant chemical categories, and including a series of nitrosamines, which have, I think Bob Joly from Eli Lilly is going to talk about this in a couple of minutes. He's been working on the relationship across a series of nitrosamines. So the carcinogenicity dose response data on the one side that we're looking at for these 78 compounds is also complicated. So there's a number of different covariates that complicate the dose response analysis. So there's a consortium of people that are working on this project now. We're actively working on this. John Wills from GSK has analyzed about 600 TGR dose response data sets. It also involves me and George and Guang Chao and David Ponting from LASA, as well as people from the HESI Genetic Toxicology Technical Committee. We're trying to grapple with some complexities related to data quality, especially on the carcinogenicity side, lesion severity, target tissue, route of administration, and treatment duration. So this complicates the analyses and basically the investigation of the relationship between carcinogenic potency and transgenic rodent mutagenic potency. But we really need to know about that relationship if we're going to proceed with this anchor compound approach for determination of acceptable intakes. Just very quickly, I wanted to talk about some work that we've been doing on uncertainty factor values. So I think many of you are probably familiar with this kind of figure, either this figure or several like it. It's from a paper by Dankovic et al.
Did we just lose Paul?
I think we might have just lost Paul. We can revisit his slides maybe when he's back online if we have some time a little later on in the agenda. Oh, there you are, Paul.
Did you lose me?
We did lose you for a second.
Oh no. What slide was I on?
You had just finished your thank you slide, and then we're moving on to I think your last couple of slides.
So this one? Can you see it now?
We cannot see your slides right now. And your video is a little choppy, so I don't know if we're maybe losing connection a little bit with you.
Yeah, I think the connection is pretty difficult. So can you see it now?
We can see not in presenter mode. Now it's in presenter mode. And
Okay,
then,
good.
Yeah, and then we are coming up on time, but
Yeah, I'm sorry about that. I'll go through this very quickly. We've been trying to look at uncertainty factors, especially uncertainty factor for inter-individual human variability, as well as for study duration. And we've been doing that by looking at how knockout of a compensatory pathway increases the sensitivity to genotoxicants. So how the BMD moves over to the left. And here I'm showing you an example for LAC-Z mutant frequency in animals that are knocked out for polymerase ADA, which is involved in translesion synthesis. There's the dose response for polymerase ADA proficient animals and the dose response for deficient animals. And what I'm arguing here is that we can look at the ratio of these BMD values to get a handle on how sensitive people can become if they're lacking the compensatory pathways that protect them from genotoxic effects. And this is work that Lorraine Gallant did when she was a postdoc. And I'm going to skip right ahead because I don't have time because of that technical glitch. But Lorraine collected data from over 1,200 data. She collected over 1,200 data sets, and we've been doing rigorous analyses to determine what would make sense as an uncertainty factor for inter-individual variability. And these are the 30 genes we looked at. I'm going to skip this slide here and just go on to my last couple of slides where we've been looking at effect of treatment duration on BMD value. So similar to what we've been interested in inter-individual variability, we're interested in what the uncertainty factor should be to compensate for treatment durations that are less than chronic. Here I'm showing you mutation frequency with error-corrected next-generation sequencing against dose in liver for animals exposed to benzobiflurantine for 28 days, 90 days, and 180 days. And we can look at the change in potency, so higher potency with increasing treatment duration, and look at the BMD ratio. So if we look at the BMD ratio for 28 days to 180 days, we get a value that's pretty close to 10, which some regulatory authorities have recommended for compensation for treatment durations that are less than chronic. We get the same range of value when we do the analyses for bone marrow. So sorry about that technical glitch, and sorry I couldn't get into more detail about the work that we've been doing on uncertainty factor determination. I just wanted to end with an acknowledgement. I didn't do this work alone. There were a large number of people from a large number of institutions. Thank you very much.
Thank you so much, Paul, for that great talk. If you have questions for Paul, please put them in the Q&A. And I would like to introduce our next speakers. So our next speakers are Andreas Zeller from Roche and Bob Jolly from Eli Lilly, who will be talking about an industry view on the use of BMD analysis and quantitative genotoxicity assessment. So welcome to you both.
Hi. Yeah, my name is Andreas, and I'm responsible for genetic tox here at Roche in Basel, Switzerland. And next slide, please. And the next one. Yeah, so back in 2015, we were developing a small molecule drug for a severe non-oncology indication. The candidate molecules were very efficacious, but our chemists couldn't get rid of a micronucleus liability. However, we could demonstrate that an indirect non-DNA reactive mechanism was responsible. We then performed 3 separate in vivo studies, each with a comprehensive TK profile. A acute combined micronucleus comet study, a 4-week micronucleus study in adult animals, and a 13-week micronucleus study in juvenile animals, as the drug was mainly intended for pediatric indications. All studies were performed in both males and females, and 2 exposure parameters, AUC and Cmax, were analyzed. Continuing the study design debate from yesterday, there are certain restrictions for guideline-regulated animal studies, and we are committed to 3R. So what we did back then was analyzing the animals of the DRF pre-study, and only then design the main study. This takes a bit more time, but I think it's a smart way of doing it. Next slide, please. So we analyzed the micronucleus response with PROST using a critical effect size of 0.5, which corresponds to an increase of 50% over concurrent control levels. That was based on my 2017 publication that uses background variability, because in my opinion, you cannot put a limit to an endpoint that cannot be measured reliably. As you can nicely see from the CEDU by CEDL ratios in green font, the data quality was excellent, way below the 5 to 10-fold cutoff that is often being used. So we are at 3, 1.4, and 1.7 here. The BMD, or rather the CED, is shown in the 5th line of the figure legends, 5th line from above, CED. And you can see they're all pretty much the same. And as you can nicely see, there was no unexpected accumulation of the drug or its effect. So the 13-week study showed actually the highest POD. So we're pretty consistent in terms of duration for this effect. And this is in contrast to what Paul just mentioned for the mutagenic effect of BBF. This is a micronucleus effect which wears out over time. It's not accumulating. Back then, we did not apply the covariate approach, but rather used the lowest of the 3 calculated BMDLs as a point of departure, POD, for micronucleus. But this is somewhat unfair to the compound, because the reason for the acute BMDL, or from the acute study on the left side, becoming the lowest, is the data variability, not the effect being exceptionally strong at the short treatment duration, as can be seen when you're comparing the CEDs instead of the CEDLs. Next slide, please. If we would have applied the covariate approach with study duration as covariate, the ranking would be slightly different. And the CEDU by CEDL ratios would also change slightly, but I'll come back to that later. Next slide, please. For Cmax, the situation is similar as for AUC. The BMDL is driven by data variability, not the CED. The CEDs here are also incredibly close together. As you can see, it's 438, 549, 649 nanogram per milliliter, actually. That's very close to each other. Next slide, please. And here is the covariate approach for Cmax, this time 470. This is where the green arrow is pointing to, would be the lowest, which is even greater than the BMD of the acute study, which was 436, and clearly higher than if you take only the individual study BMDL, which was 236. The difference of these 3 individual assessments versus the covariate approach could be related to the normalization, which I did for that purpose, or simply because of the, say, violation of the covariate requirements, as one would then be assuming that, for example, the adult and the juvenile rats would behave exactly the same. I'm sitting on the fence with regard to the covariate approach here. Next slide, please. So here's a bit of a summary and some recommendations from my part. So first of all, BMD works and it's acceptable for health authorities. Secondly, BMD is clearly superior to the NoAEL approach for a couple of reasons. However, in this present case, both would have basically ended up in the same place. Third, the data quality is key, not only in terms of low variability, but also with regard to the number of dose levels. And number four, in my opinion, mechanistic evidence like fish staining, etc., is certainly appreciated by health authorities. And to close down my part of this talk, our predictions have meanwhile been confirmed in 2 separate negative carcinogenicity studies with this drug. And now I'm passing on to Bob.
Can you hear me? Okay.
Yes, we can hear you.
Awesome. So I'm going to start with just some general information. We're going to go back to just industry perspective or pharma perspectives on use of BMD. I'll then move to some of the challenges I've seen in using BMD for quantitative gen tox, but then follow up with some of the nitrosamine work that's been alluded to and actually shown. So, I'm sorry. Can you go back one slide? I didn't hit that. So historically, the BMD analysis of pharma has been pretty limited, and a lack of broad HA acceptance. You've seen some of the differing approaches that are out there, the having to justify what BMR or CES you're going to use, that sort of thing. But also just that standard tox designs aren't really suitable for analysis for BMD analysis or curve fitting, or really tend not to have a large number of groups to really detail the curve. Now that might be just my bias, because I think many folks would say you can still do the BMD on those standard study designs, and you can still get better quantitation relative to what we do now, which is no gel, no L type approaches, as the BMD in general uses the full dose response. But some of the challenges of doing that have been highlighted in previous talks. I think the nitrosamine work that I'm going to show at the end of this talk or case study really illustrates that there may be opportunities to widen the use of BMD approach for quantitative quantitative assessment for genotoxicity, both using TGR and possibly ECS. Next slide, please. So, we're going to rigorously follow 488 when we do these. We might be looking at an increased number of groups for dose characterization versus risk characterization versus hazard ID, which would generally be 3 groups. We might go to 4 to 6. Whether you can reduce the number of animals certainly seems like you might be able to, but there might be challenges there as well. Identifying the BMR and CES is critical, and you've seen the work that supports that done by Paul and George and others, and there's a really rich literature supporting what values should be used for the BMR for the TGR data, right? And we're reliant on that, and that sets the stage for doing this work in a consistent way. The tools that are available, people mentioned it, Prost, is really easy to use. I can vouch for that because I do it, and if I can do it, anybody can do it. But the R scripts are there as well that give you a little deeper dive into graphing and additional quantitation. I know there's an IU site for Bayesian assessments. We generally haven't done that because the literature isn't really, at least the literature for the quantitative gen tox that I'm going to show you doesn't really seem to support that one way or the other. But the model averaging and the bootstrapping appear to be really robust and really do make the ability to do the BMD a lot easier from a layman toxicologist view. Next slide, please. So this is the literature. George alluded to it yesterday. There's this rich history. It's like we're standing on the shoulders of really good scientific work by really good scientists in this particular area. So we've built on that with some of the nitrosamine work I'm going to show you in a bit. But before I get to that, next slide. So this is some of the challenges I've seen in doing quantitative BMD analysis for GenTox. This is mutant frequency TGR data in the liver for a molecule on the left that had poor fit and a molecule on the right that had really good fit. And on the left, that firstmost graph there, there is a trend towards, and there's actually this is low, medium, high kind of design, so it's only in effect at the high dose, and that was discussed as being problematic in terms of fitting earlier today. But it was statistically significant at the high dose, but at no other doses. And you can see it's a very variable data set. It's not a good fit, right? And so it had some outliers. There were some hot spots present in the data. When those are removed, you get a better fit. But and I don't know that it's easy to see, but the overall response actually was much reduced when we removed the outliers. It went from a log 2.4 to a log 1.8. And you compare that fit to the fit for the nitrosamine, nicotine-derived nitrosamine, which is in the data set I'll be showing in a little bit. And maybe you can't see the dots, but we do have multiple points on the curve that had response that weren't max, and we treated to an MTD in that study. But you just have a very tight dose response, and the model averaging is very tight. So if you go back to the one on the left, the overall the range of the BMDU to the BMDL is like 30 to 40x in that, where you kind of want to be less than 5-ish is a really good fit in my estimation. Again, just a toxicologist, not a modeler here. Whereas when you move the outliers, that actually that range goes down. It's I think on the order of 10x, but for the NNK data, the range is like 3x. So the key point here is, yeah, you have this positive response, but the criteria for the study, and this was a hazard study, was that the data, it was a negative molecule because it didn't exceed 2x, and it definitely didn't exceed the historical data in liver, right? So I think it's still important to consider the context of those data and what your criteria is for a positive response, even when you're doing the quantitative assessment. And we've seen this kind of response. I've seen the response where it was the mid dose that was statistically significant, not the high dose. And I've seen more troubling where the control data, even in validated tissues or tissues that have a good historical set, where the control data are either low or high, and that really messes up your study. And it's just a challenge when you're interrogating the low end of the curve, which is one of the strengths of BMD. Where do you see a positive response? And you have that kind of flux. But in general, we've had really, really good data and reproducibility, but these are challenges. I mean, biology is never easy in terms of what the endpoints are. Next slide, please. So just based on that, what's our view on mechanism-based risk assessment of genotoxicity? We think it's a warranted alternative relative to linear low-dose extrapolation based on CAR-C data. I'm really thinking about the data I'm going to show you in a bit, the nitrosamine data, where the mutation data is clearly relevant and a sensitive endpoint. It's acknowledged to be a precursor. We know a lot about the mechanism for those molecules. And then the final bullet is just that the transgenic assay is very robust, well validated OECD assay. So we feel pretty strongly that this is a potential way we could use data. To Paul's point, it hasn't been accepted, but it's something that we're discussing. So on to the data. Next slide, please. So Paul showed this. George showed this yesterday. This was published recently. This is the summary of the summary paper from the in vivo nitrosamine HESI group work, and I think it was published a few months ago, finally. And really, what you're seeing here is we took 7 nitrosamines that had robust CAR-C data, released defined by Berkey et al. In 2023, 2-year duration, multiple dose groups, both genders, etc. But they were molecules that had robust CAR-C data where the TD50 value was considered to be a robust value. And then the different partners in the HESI group ran these in the TGR studies, 28-day TGR studies with 3-day fixation. And this is mostly liver data. There's a couple examples. NNK is an example where we have liver and lung. And then this is rat mutant frequency, but there are a couple examples where there's mutant mouse and big blue mouse as well. But as you can see, the correlation of the BMDL50, again, we're using the value based on the literature that has been provided for this methodology, the BMDL50 correlated incredibly well with the TD50 data, right? And the R squared was 0.95. So 95% of the correlation of the variation of these two variables is captured in the graph. The Pearson's value is 0.97. So there's an incredibly high correlation for these data, for this data. And then I think Paul was mentioning that, he's expanding this to other structures and other compounds, and that data is hopefully going to be forthcoming in the next year or two. Next slide, please. So these are the BMDL values that were calculated. All of these molecules were published independently, and then we publish as a group. So if you're interested in the overall paper, it's on the citations on the right there. But each of the contributors publish these independently as well. But the point is that the BMDL values correlate really, or essentially lay on top of the gold TD50. They're not always higher or lower. It goes both ways, but essentially they're good estimates, a good surrogate of the TD50 value. And while you're talking 1 month versus 24 months, and you might expect the BMDL50s to drop over time, and I don't necessarily expect that it would be more than 10x or 20x, and these values sit right on top of the 2-year data. Next slide. And so this is a representation of the confidence intervals for those same 7 nitrosamines. And really what it's showing is that the assay as is can really delineate between really potent nitrosamines less than TTC of 1.5 mg per kg versus slightly less potent nitrosamines greater than 1.5 mg per kg. I mean, we would have loved to have more nitrosamines with robust CARSI data, but these are the molecules that fit that criteria, and this is and they're classic molecules that are used for comparison and read across. Next one. So summary: we think the BMD value really does provide a better evaluation of potency, whether it's for gene tox endpoints or anything. It uses the entire dose response curve. It has value. Being able to do it consistently and in a consistent manner, it really does. And this is a case with the Gentox data where you can do that. Really does help the interpretation and the communication. It can provide a POD for non-genotoxic endpoints, but here for the nitrosamines, we're really using this data as a surrogate for CARSI data because we're not going to be able to run CARSI studies on these low-level impurities. It's just not going to happen. We need some way to evaluate this. This is a possible way to evaluate these and get a better estimate for the potency of these molecules. And again, the one point that needs to be reiterated is we're using the BMDL, the lower level of the confidence interval versus the TD50, the central metric for the CARSI, but we're also applying a very large uncertainty factor to this, just like we would for the TD50 data. So we'd follow, if we were to use this kind of approach, whether we'd use that curve to do a direct extrapolation or other methods that are described in the paper, the citation I provided, you are essentially doing a similar type assessment and you're going to land in the same place. So our argument is this is a valid type approach, a good way, a good surrogate for the CARSI data, and it would give appropriately protective limits. So I think that's my last slide. So thank you for that. And sorry I rushed through, but there you go.
Thank you so much. Thanks, Andreas and Bob, for that great talk. We had a little bit of a change in our agenda today. So our next speakers will be Fabrice Neslany and Romain Grahl from L'Oreal, who will be talking about BMD for cosmetics today, promise meets uncertainty. So welcome to both of you.
Yes. No. Cannot hear me?
We can hear you. We cannot see you, and your slides are not in presentation mode yet. But...
okay. So I have to check how to modify. You can see me? No?
We can't see you, but I think the bigger thing is just putting the slides in presentation mode. We can handle the camera being off.
In this distance, I'm sure it's a pity. I don't try previously, sorry. Okay, maybe if you have my slide, maybe it's simple that you show my slide.
Yep, I can do that.
I don't find the presentable model. Thank you, sorry for that.
No problem. We'll switch and share slides on our end. Just hold tight for 1 second while we get those pulled up.
Thank you. Okay, thank you. I can see. Sorry for this technical issues. So thank you for your invitation and giving us the opportunity to make this presentation with my colleague, Romagal. So next slide. Yes, thank you. First, it is important to remind the context of the animal testing ban in Europe for cosmetics. We have two key dates, which are 11th of March 2009 and 11th of March 2013, with, as you can see, successively a ban on the use of animals for assessing acute and local effects, as well as genotoxicity. Then in 2013, a total ban on the use of animals for repeated dose toxicity studies, including developmental and reproductive toxicity. Next slide, please. Yes, so the problem is that systemic toxicity plays a determining role for safety assessment, for safety evaluation. We need for a POD derived from systemic effects to calculate, next slide please, the margin of safety. The margin of safety corresponds, as you can see, to the systemic POD, either the NOAEL or the BMDL, divided by the estimated systemic exposure dose. And each ingredient in the final product is used at a concentration at least 100-fold below the concentration that produce adverse effects. Next slide, please. So as mentioned before, systemic toxicity includes DART and repeated dose toxicity. And for both, if in vivo studies complying with animal testing ban provision, that is to say, carried out before March 2013, if they are available, they can be used for risk assessment. And in such cases, the BMDL can be calculated and used. If no, DART and repeated dose toxicity studies are available, read-across assessment combined with a weight of evidence approach can be used. And the same way, BMDL or the analog can be calculated and used. The last case, if no studies, no read-across is working, we move towards full NAM methods and NGRA. NGRA is defined as an exposure-led, hypothesis-driven risk assessment approach. It is mainly based on NAM's new approach methodology, in vitro methods. And for such in vitro studies, questions arise regarding the use of the BMDL, its relevance, the parameters that will have a direct impact, and so on, that we will present you. Next slide, please. You can, yeah, next click, please. Yeah. To perform NGRA and how to calculate the BER, bioactivity exposure ratio, generic or specific assays are needed. Next click, yeah. The point of departure, POD, either the inhibitory concentration or the BMDL, is estimated from the generic assay or from specific assays relying on general toxicity, specific biomarkers, targeting mode of action, key event of LP, and so on. Then, human exposure is estimated from PBPK modeling, allowing to calculate the plasma concentration. And finally, either the BER or the MOS can be calculated by dividing the POD by the systemic exposure. We can see here the key role played by generic and specific in vitro models, which I would like to explain in a little more detail before moving on the calculation of BMD. So please, we can, next slide. The first, the generic assay. It has been reported that off-target interactions of pharmaceutical drugs with some tissue macromolecules can cause numerous clinically important human adverse drug reactions. An in vivo pharmacology profiling panel has been adapted for evaluating the activities of cosmetic ingredients as published by our colleague Matthew Burbank. Next slide, next click, please. Yeah. This panel comprises 79 targets, including G protein-coupled receptors, nuclear receptors, enzymes, ion channels, and transporters, assessed using 83 assays, including binding assays, enzyme assays, and cellular assays. Next slide, please. Transcriptomic analysis techniques are used to study the expression level of all transcripts, that is to say, at the RNA level. New sequencing methods are performed, allowing the whole transcriptome gene expression profiling. The initial proposal, as published by Middleton in 2022, is to conduct transcriptomic studies on 3 cell types. Here you can see FG2, MCF7, and EPA-RG after short or continuous treatment across a wide range of concentration. And as a result, the differentially expressed genes, DEG, that is to say, those genes that significantly change between a control and a treated group, are determined. Next slide, please. Then we can leverage on the information derived from these results and apply different levels of interpretation. Once a list of DEGs has been identified, the next challenge is to move from this list to a biological meaning. This process is known as functional annotation, which can use different methods to determine which pathways are activate or deactivate. We can use, for instance, the overall representation analysis, or RA method, or the gene set enrichment analysis, GSEA. From DEG, you can see that it is also possible to calculate a BMD based either on gene level or on the pathway level modulation. Next slide, please. Finally, specific assays can be performed on 2D or 3D models, such as spheroids, organoids, or organochips, on which viability and modulation of specific biomarkers can be assessed. For instance, in this case, for this liver 3D models, viability and albumin secretion were quantified, and lethal and inhibitory concentration 20% were calculated. Now I will give the floor to my colleague Romain, who will present BMDL calculation cases and the sources of variability. Please, Romain, the floor is yours.
Romain, we can't hear you.
Can you hear me?
Yes, now we can. Thank you.
Oh, I was sorry. So I start over. So here is an example coming from a real case study of cosmetic ingredients for which we've been performing several tests and calculated the BMDs or equivalent. And first few comments on that. We have, as presented, various level of complexity regarding the experiments or the cell models, but also in terms of readouts, because there is some readouts that are quite physiological relevant with the albumin quantification, for example, in more complex 3D assays. But also for the transcriptomics, we are at the gene level, and at the gene level, when you calculate the BMDs, sometimes it leads to maybe two generic results, and it's much more the same result when we go at the pathway level. Here we have an example where we got at the pathway level 0.53 micromolar on the trabecular formation pathway, which is not very meaningful for us. But maybe it becomes more interesting when combining both the result from the BMD calculation and the biological interpretation. It means that just keeping the lowest calculated BMD, considering it as the most conservative, might miss the biological and the toxicological considerations. So we've been through the question of the BMD calculation, given the heterogeneity of our data, to improve the confidence and the biological relevance for a robust safety evaluation. So next slide, please. So everything starts from the curve fitting, and we propose to take advantage of already existing approaches like the BMD Express that have been developed by the SEPA, where multiple mathematical models run in parallel, and the most performant one is kept. It avoids a one-fits-all model that might introduce many uncertainties given the heterogeneity of our data. Next slide, please. So then we have explored several parameters of our experimental setups that might influence the BMD calculation, like the number of concentration, the number of replicates, the variability, and the outlier management. We've been working on a set of 50 samples with dozens of permutations to increase the statistical relevance. So next slide, please. So the first is the number of concentration. We have performed dozens of permutations, removing every replicate of one concentration and evaluating the impact on the calculated IC10. You can click, please, to get the example. Yes, thank you. So obviously, by removing concentration, we observe an impact with the greatest impact when removing one of the top 3 concentration. And you can, no, it just come back before. Sorry, no, at the concentration, it was for the previous one, just, because we have a modification when we remove the concentration, and especially when we remove the top 3 concentration, which has an impact on the design of the experimental setup, the defining the concentration range. We can go to the next slide, please, and click maybe 2 times. Yes. And yes, thank you. Sorry for the emission. Regarding no, we have to come back because we are at the variability. Yes, thank you. Regarding the replicates, given our data set, we observed that 1/3 of the condition have IC significantly impact when removing 1 replicate and 2/3 when removing 2 replicates. Thus, the recommendation would be to increase to at least 4 replicates to increase confidence in the calculated BMD and to allow some robustness into the calculation. So you can go to the next slide, please. Okay, thank you. You can click 1 time. Yes. Removing considering the variability, we observe the huge impact that reinforce the fact that controlling the robustness of the experimental design is key to building confidence into the result of the calculated BMD. And you can go on the next slide, please. Okay, you can click maybe 1 time, 2 times. Thank you. So finally, for the outlier management, confirming that it has also a great impact on the calculation with 2 recommendations here. The first is the importance of filtering out the outliers, because when we see that when we remove the outliers, we have a great impact on the IC calculated. But also having enough replicates allow us to remove with good confidence the different outliers and that have an impact on the BMD that is calculated. So come back to the number of replicates, which is maybe the most important fact on that topic for the BMD calculation. And we can go on the next slide. Yes, thank you.
So to conclude, thank you, Romain. So to conclude, so just on the context, so since 2013, animal testing has been banned. in Europe. But we need for toxicological reference value on repeated dose toxicity for calculating the margin of safety. BMDL is potentially usable for in vivo studies complying with animal testing ban provisions, either on the ingredient or on its analog if the read-across is working. In absence of data, it is possible to calculate BMDL on results from generic or specific in vitro assays, but we face issues regarding the heterogeneity of data from current in vitro tests, as Romain just showed you. The heterogeneity of the data relates to the number of concentration of biological replicates, to the variability, and to the outlier handling. But beyond the metrics, inhibitory concentration, BMDL, BER, the biological relevance must be considered. For instance, just one example, the calculation of a POD from some bioactivity data, specifically transcriptomic data, is still under debate for us, especially when considering the value using the gene level POD or the pathway level ones. Thank you very much for your attention, and sorry for the technical issues.
Thank you so much, Fabrice and Romain, for this great talk. As a reminder to everyone, please ask questions in the Q&A box, and we will get to those in written form or during our Q&A session at the end. But with that, I would like to introduce our next speaker, who will be Guangchao Chen from RABM, who will be talking about the derivation of benchmark responses for hematological endpoints. So welcome, Guangchao.
Yeah, thank you, Claire. I'm Guangchao Chen from RVM. I think the topic of choosing a benchmark response has been brought up quite a few times throughout the whole workshop. So by this presentation, I would like to propose one of the solution to choose benchmark response for a continuous endpoint and using hematological endpoints as a case study. So this is actually about the work that we have submitted. Due to the limited time, I have cut out a lot. So I hope that is still clear enough. So when we do the benchmark dose analysis, three major choices, including choosing BMR, then choosing the models to fit your data, as well as choosing the value to use as your point of departure. So as many speakers already brought up, model averaging is the recommended approach to use in this case. It doesn't matter what tool, as long as you choose one tool. I think the approach model averaging is already well automated. So that issue is more or less already solved. Meanwhile, I think we all know that we use BMDL as a point of departure to go further with our chemical risk assessment. But the choice of benchmark response for a particular endpoint, I think there's still a lot of discussion about those. So today I will show some ideas how we could handle that. Next slide, please. When it comes to a continuous endpoint, for instance, when we talk about the choice of BMR, as we already saw before, the first thing we need to deal with is the definition of the BMR. What kind of the matrix? For instance, in the absolute sense or in the relative sense. And also in the relative sense, it's like additive or multiplicative, the ratio. And we already know that as recommended, for instance, by EFSA, we use the percentage, which is multiplicative BMR. The second question in this case we need to answer is, okay, we choose a percentage, but then how much percent? If 5 or 10 or 1 or 20 for a lot of endpoint is still not known. So this is something we still need to deal with. Next slide, please. To handle this situation, Paul actually already touched upon, thanks, so it saves me some effort. What Slop proposed is theory called effect size theory to handle this. The theory is quite straightforward as I see how to derive BMR for a particular continuous endpoint. In the figure, on the right side, we see a dose response for continuous endpoint. And then we see that the background, and we see that the curves levels off. And then on the log response scale, the difference between the background and the plateau, it is the log of the maximum fold change of the response. Throughout this presentation, I will just call it M instead of maximum fold change of response, which is easier. So the distance is log M. In the effect size theory, Wild basically proposed that let's just take a portion of this log M as the endpoint specific BMR. For instance, it proposed that 1/8 of this log M, which is, if you look at that in the original scale, which is the M up to the power of 1/8, we can see this as a very small effect, and then 1/4, for instance, medium effect, and 1/2 of those, 1/2 is a large effect. So that is proposal. This BMR, of course, is in fold change, and when we want to have a percentage, we can very easily calculate the percentage number based on the fold change. So the idea is really easy. Just estimate your maximum fold change in response, estimate your M, take a proportion, for instance, 1/8, and you get your BMR in fold change for the endpoint. But then some people may start thinking, well, all nice and simple, but a lot of times, the data set, actually, we do not have much information at the higher dose range, like where the dose response should level off, which is true. And then there happened to be one more very nice discovery in the paper, which is in the next slides we can see. Well, the next slide, please. Yeah. So Wild actually analyzed the relationship between the standard deviation, which is the square root of the variance, and the M, maximum fold change response. And then in the figure, you can see there's a quite nice relationship based on 27 endpoints of in vivo toxicity studies. And then with the equation, and that is how a standard deviation and the maximum fold change are related. So the M equals to e to the power of Z and standard deviation S. And in this case, based on the data at hand, S is around, Z is a constant, which is around 7 with confidence interval. So basically, what we're saying here is when the standard deviation or the variation is larger than the maximum fold change in response, the M is also larger. I think a lot of people see this and they probably have the idea, oh, actually, it makes sense. It feels right, yeah? Like some people may think, when the natural variation of a certain endpoint is large, and then a lot of people will feel like, oh, maybe actually the M, maximum fold change of the response, also larger. That is what we're showing here. But then why is that? Could we go back to the previous slides, please? So there's an explanation, actually. So in the figure, we now see a relationship between the dose and the response. So in this case, we're interested in the change of dose as a factor, how that influenced the change in response. That is true. But actually, in the experiment, there are also other factors in your experiment that can influence your outcome, can also influence this response. Based on assumption, we think, okay, the other factors, they are either constant or they are perfectly randomized. But in reality, it will never be the case. So let's say when M is larger, and then in this case, I will call that endpoint is more expressive to the dose. So that one endpoint is more expressive to the dose. Then we think, then it makes sense that that endpoint will be more expressive to the influence of other factors as well. Because in nature, dose is just one of the factors in your experiment. And there are also other factors that over there. And the nature wouldn't care if it's a dose or other factors, being that the sensitivity of the animals or exposure duration or whatever factors might influence your outcome. It's all the same for the nature for biology. So that means when the endpoint is more expressive regarding the dose, then it should be also to other factors in your experiment. And in what kind of situation do we need to deal with the influence of other experimental factors on your outcome, then we should look at the variation of your dose response. So that is actually also what we discovered. So there's a correlation between your variance and your maximum fold change of response. Next slide, please. Yeah. And then here, like I said, just give you go back to what the proposal. So we can derive BMR based on a proportion of the log m or m to the power of alpha, and then we say 1/8, for instance. We can see that as a small effect, and then when we cannot estimate the m rather precisely, we can use standard deviation, which is square root of variance, as a surrogate. Could we go back, please? Previous slide. Yeah, exactly. And then we can use a standard deviation as a surrogate, and then we can derive the BMR as well. And if you look at the equation on the top, actually, more or less, you can see we are more or less approximately taking the one standard deviation as well. So two things I want to point out here. So here, the one standard deviation is not just for the control group, it's the entire dose response, because for the continuous response, assuming a log normal distribution, then on the log response scale, the variance should be constant. So we're using all the dose group information to estimate this variance and therefore the standard deviation. So that's one thing. And the other thing is, so these two, they are numerically actually agree with each other more or less. And then this is the advantage, as I see, of this theory, because it reconciles the two matrix we talked about before, like yesterday, Lynn brought up the 1SD matrix and as well the percentage BMR proposed by EFSA. So they both have pros and cons. For instance, 1SD taking into consideration the natural variability of one endpoint. But meanwhile, because it's the standard deviation, so different data quality will give you different BMR. For instance, a good quality data with a small standard deviation, then you get small BMR. But then when the data quality is bad, then you get a large BMR. But then by definition, the BMR is actually a response that in chemical risk assessment we consider as non-adverse or acceptable as such. So the answer to this kind of question obviously should not be dependent on your data quality. So that's just not logical. Meanwhile, the percentage BMR does not suffer from this issue. But as well, like Lynn mentioned, the same percentage applying to a different endpoint also doesn't sound really logical. For instance, 10% body weight decrease and 10% brain weight decrease, they should not be seen as equally severe in chemical risk assessment. So when you use the effect size theory, basically, it's telling you when the maximum fold change is larger, when the natural variability is larger, then your benchmark response should be larger as well. So meanwhile, so it considers natural variability of the endpoint. Meanwhile, it applies to different endpoint, and you can derive different percentage and not just like a same percentage for all. So I think that is the advantage of this theory, and it reconciles the two matrices we've been talking for quite a while previously. One thing that should be noted is that so this variance as well as the standard deviation should be derived based on large amount of data. In this case, we have more confidence to think that this variance and standard deviation is more representative to the natural variability of that endpoint. And when we use a single data set or very few data sets, there's a higher risk that you will get a bad indication of this variation standard deviation of that endpoint. Yeah. Next, please. Next slide, please. Just to be short, how to derive benchmark response based on this effect size theory? 2 steps or 3. Firstly, you perform your BMD analysis based on a large amount of data for the endpoint you are interested in. Afterwards, you derive the maximum fold change of response, and then you can scale your BMR based on that. And alternatively, you can also derive the variance and get your standard deviation. and then use the equation using to derive the BMR as well. Next slide, please. Here's one case study I want to show. So we gathered a lot of data from the NTP database about hematological endpoint. There are a lot of records you can see here, and then most of those studies relate to rat and mouse, and they all animal studies. The data set we got contains a lot of chemicals and also a lot of endpoints. Could we go next, please? But not all the endpoints are relevant here. So we selected the endpoint that are relevant for chemical risk assessment. So there are three groups. First group is the endpoint related to a red blood cell. And you can see what are those, and then what do they mean, as well as what their indication in the sense of toxicology. And then a second group is white blood cell count, and third group is platelet counts. Yeah, could we go next? Yeah, so here's the results. I'm only showing the red blood cell, white blood cell, and platelet, and then a number of the data set, and as well the calculated variance standard deviation estimated maximum fold change based on the standard deviation, as well as the benchmark response. If we are going to use the equations I just showed you and how much the BMR would be. And here we can see actually, for instance, for the red blood cell count, the BMR is rather small. The variations themselves are really small and narrow. And then for the white blood cell, is relatively larger than that, like around 20%. And for the platelets, is like somewhere in the middle, like 10%. And then I purposely put the figures on the right side as well, so that you can also get a feeling. For instance, you can see for the red blood cells, the variation really narrow. And then in the middle is white blood cells, the variation is larger, and platelet somewhere in between. And here I would like to point out one thing, which is the power use a large amount of data set, so that I do feel like sometimes some people might have the idea that the data is the reality. So I have the data, I do the analysis, the data tell me something that is the case, the data don't tell me something that is not the case. If I see effect of a compound, that is safe. If I don't see effect, then there's no effect. But the fact is actually your data contains part of your data contains part of the reality, plus some errors, sampling errors and measurement errors. So that's why when we, for instance, analyze single data set, there's always a higher risk. You get a bad indication. And then here we put all the data together and we use all the power of the data and analyze one variance. And then if you look at the figure, for instance, in the middle, I think a lot of people would get the impression like, oh yeah, the variation of the white blood cell count is like more or less larger than the platelet and certainly larger than the red blood cell. And then you can also, in this case, look past the noises in the data set. And then if you only have a single data set or a few data sets together, then I think there's a higher chance your indication will be biased. Yeah, so that's what I want to point out here. Could we go next, please? So there are also other endpoints related to red blood cell. So here you can see hematocrit and hemoglobin and others. And then based on this calculation, actually, the BMR is also pretty low. So basically, our conclusion here is the endpoint related to the red blood cell counts, their variation is very small and narrow. Can we go to the next slide, please? I think look past the numbers, of course, there are also some biological explanation of why is that. For instance, the function of red blood cell, is to transport oxygen into the tissues and then transport the carbon dioxide out. All the process is very regulated in the body. The turnover, the cells, generation and clearance of the cells, so it's pretty tightly regulated. Meanwhile, a white blood cell, the function is to define the body against foreign organisms. And for platelet, it's very important that they aggregate and clot to stop the bleeding. So for white blood cell and platelet, it's also reasonable that those endpoints, they vary largely. Next slide, please. I think that might be my last one. So based on all the calculations, so we propose finally for red blood cell and endpoint related, we use 5%, and for platelet it's 10%, and white blood cell count 20%. I believe that's the last slide of my presentation. Yes.
Thank you so much, Guangchao, for that great talk. I would like to introduce our next speakers, Omari Bandele and Laura Markley from the US FDA, who will be talking about benchmark dose modeling for food chemical safety, perspectives from FDA's Human Food Program. So welcome to both of you.
Thank you. I want to just check and make sure you can hear me.
Yes, we can hear you.
Great. So first, just thanks to HESI, the organizing committee, for inviting us to be a part of this workshop. Again, I'm Omari Bandele from the FDA's Human Foods Program, and I'm very pleased to be here with my colleague, Laura Markley. Today, we'll be discussing the role of BMD in food chemical safety, focusing in particular on perspectives from the Human Foods Program, or HFP, as I refer to it on my slides. I'll begin with a brief introduction to HFP and share some general perspectives on how BMD is used as a tool in our safety assessment toolbox for regulatory decision making. My remarks will stay at a high level and will not go into the technical details of how BMD is applied or evaluated in HFP. And I also want to acknowledge that some of what I'll cover will reiterate points that have already been raised during the workshop. Laura will then take you through a case study that illustrates how BMD was used to support a safety decision in the review of a food contact substance. So next slide. Before I start, I just want to make clear that the views we're presenting are our own views and do not necessarily reflect the official views or policies of FDA regarding BMD. Next slide. Okay, so this chart here, it shows FDA's organization at a high level, and I want to highlight that the 7 units most directly involved in regulatory decision making. Each of these units plays a distinct regulatory role. But today, our focus is on HFP, FDA's dedicated program for human food safety and nutrition. HFP oversees the agency's activities related to human foods and supports risk-based, science-driven decision-making aimed at helping to ensure the safety of the nation's food supply, including the safety of chemicals in food. So next slide. This figure, it shows the breadth of HFP's regulatory landscape. At a high level, FDA regulates roughly 80% of the U.S. food supply. The categories on this figure illustrate how broad that responsibility is. HFP's work includes direct and indirect food additives, color additives, GRAS substances, nutrients, contaminants, and dietary supplements. Together, these areas reflect a wide range of food chemical safety decisions made under different legal authorities and regulatory pathways. That work also spans pre-market and post-market assessments. So in some cases, HFP evaluates substances before they enter the market, and in other cases, it reassesses chemicals and exposures as new information becomes available. It is also important to note that HFP's role is not limited to substances intentionally added to food. It also includes contaminants, so including chemicals that can form during manufacturing or processing, as well as chemicals that can enter the food supply from the environment through soil, water, or air. Dietary supplements are another important part of HFP's regulatory landscape. So FDA regulates dietary supplements as a category of food, and that includes not only the finished supplement product, but also its dietary ingredients. So the main point here is that HFP operates across a wide and scientifically diverse regulatory landscape. And because these areas involve different legal frameworks and decision contexts, the role and application of BMD can differ across product areas. So next slide. Next, I want to discuss how BMD currently fits within HFP's regulatory decision-making processes. The main point is that HFP does not have guidance specific to BMD, and BMD is not a required method for deriving a reference point or point of departure in our safety assessments. HFP generally works within a broad science-based weight of evidence framework that is consistent with the principles reflected in FDA's Red Book. Decisions are informed by the totality of the information or the evidence, including toxicity data, exposure assessments, the relevance and quality of the available studies, and scientific judgment. Within this framework, HFP considers other approaches that may support food chemical safety evaluations, and BMD can be one of those approaches. Where appropriate, it may provide a useful way to characterize dose response information and inform the broader assessment, but it is not a requirement for regulatory decision making within HFP. So in practice, the use of BMD is considered on a case-by-case basis, taking into account whether it's scientifically appropriate for the available data set that is being evaluated and the operational whether it's operationally practical in the context of the specific regulatory question that's being asked. So go to the next slide. At a high level, the Red Book, it provides a framework for the type of toxicological information HFP recommends, and this includes how studies should be designed to support food ingredient safety evaluations. So historically, many toxicity studies used in food ingredient reviews were developed with Red Book recommendations in mind. And these studies are typically intended to support hazard identification and overall safety evaluation of food ingredients. So therefore, the Red Book, it was not developed specifically to support BMD, and it doesn't include recommendations on study design with BMD optimization as the primary objective. Instead, it focuses more broadly on the overall toxicology package needed for the safety assessment. So this matters because some existing food ingredient data sets may not be ideal for robust BMD analysis. Some of these, again, these are types of points that have already been discussed in certain contexts earlier. So for example, the number and spacing of dose groups may not give the kind of dose response resolution that makes BMD straightforward. And some endpoints may be better suited than others for fitting a BMD model. So the issue isn't that these studies are inadequate for safety review, rather it's that they may not have been designed with BMD in mind. And from a HFP perspective, safety decisions have often been made effectively using a broad protective framework that incorporates conservative exposure estimates that consider sensitive populations and high intake consumers, the available toxicity data, uncertainty factors, and where appropriate, limitations on the use of a particular substance. So this overall approach has supported food safety decisions over time, even though newer analytical tools such as BMD may be used in some specific contexts. It may be seen as viewed as useful in some assessments. So the main takeaway here is that Red Book informed studies remain highly relevant to HFP's work, but they aren't necessarily designed for using BMD to determine a point of departure or reference point. Its utility will depend on the composition of the data set, the endpoints being considered, and the specific regulatory question HFP is trying to answer. So next slide. So I do want to provide a brief example or a few examples of how BMD has been applied within HFP. In this case, FDA scientists combine BMD with physiologically based pharmacokinetic modeling to derive an oral toxicological reference value range for cadmium. The TRV can then be used in dietary exposure assessments to help characterize potential health concerns from cadmium in food. So this example illustrates 2 points I've covered earlier. First, the cadmium assessment relied on multiple lines of evidence, including a prior systematic review, human epidemiologic data, PBPK modeling, and supporting animal data. Second, it illustrates that HFP uses BMD when the data supports it and when it adds value to the assessment. Next slide. All right, so looking ahead, new approach methodologies or NAMs may create more opportunities for BMD-related approaches in HFP. FDA is modernizing how it's evaluating chemicals in foods, and this includes considering additional scientific approaches such as BMD. Some NAMs may provide richer concentration, response, and mechanistic data compared to data available from some legacy toxicity studies. And these data sets may be suitable for benchmark concentration approaches. And as NAMs continue to mature, this could expand the use of BMD-related methods for food chemical safety assessments in HFP. However, the use of NAMs does not automatically mean that BMD should be applied. First, it's important to determine whether BMD is appropriate for the regulatory decision. Next slide. I'm going to finish my part here by providing some additional examples of how BMD approaches have been applied in HFP research using NAMS data. In one example, BMC analysis was applied to transcriptomic data from human induced pluripotent stem cell induced hepatocytes exposed to silver nanoparticles to derive transcriptomic points of departure. A similar approach was applied to cannabinoids in human induced pluripotent stem cell derived hepatocytes. In that work, concentration response transcriptomic analysis and BMC modeling were used to compare relative potency across compounds and to support deriving transcriptomic points of departure. And the last example, Bayesian BMD modeling of cell viability data from human testicular Leydig cells was used to compare the potency of cannabidiol and its main metabolites. So taken together, these three examples show that BMD-related approaches can be applied across different NAMS platforms, endpoints, and questions. So the key takeaway here is that BMD is already being used in HFP research. And while these examples here represent research applications, they highlight the potential broader role of BMD-related approaches in food chemical assessments as NAMS continue to evolve. So with that broader perspective in mind, I will turn it over to Laura, who will walk you through a more detailed example of how BMD was applied during the review of food contact submission.
Great. Can you hear and see me?
Yes.
Yes. Yes.
Great. Okay, thank you, Amari, for the excellent overview of how BMD fits into the FDA's Human Foods Program. And thank you to the HESI team for organizing this very informative two-day workshop. I'm Dr. Laura Markley. I'm a regulatory toxicology reviewer in the Office of Pre-Market Additive Safety. This case study that I'm going to share with you today involves a food contact substance, so not an impurity, but an actual food contact substance itself. And it was an assessment from a real-world challenge that we came across, and we encountered a challenge where the traditional Noel approach was giving us difficulties, and the benchmark dose modeling helped us validate a defensible point of departure for degradation of the acceptable daily intake. So I performed this analysis with my colleague, Dr. Jeff Patton, who is now retired, with support from our then supervisor, Dr. Omari Bandalay, and we received valuable guidance from Dr. Alan Davis at EPA. This work was conducted about 5 years ago, but it still remains a great example of how BMD can support regulatory decision-making in practice. Next slide, please. So first, let me provide some context while keeping the substance anonymous. We were evaluating an anti-static agent, and it was intended for use in food contact materials that would come into contact with infant formula in human milk. So this is a particularly sensitive population of concern. The notifier had already submitted 2 subchronic toxicity studies that are shown here in the table. In the 90-day rat study, the NOEL was 15 milligrams per kilogram per day, with effects at 30 milligrams per kilogram per day, including mortality, respiratory effects, cataracts, and decreased body weight. In the dog study, the NOEL was higher at 30 milligrams per kilogram per day. Notably, though, the rat study showed cataracts at the low L, so this will become important later. Next slide. Then the notifier provided us a new toxicity study, a prenatal developmental toxicity study in pregnant female Worcester rats. This was a well-conducted study with no major deficiencies. And then as depicted in the figure on the right, the rats were dosed by oral gavage at 0, 10, 30, or 90 milligrams per kilogram per day during gestation days 6 through 20, which is the critical window of organogenesis. The study authors reported a NOEL for developmental toxicity of 10 milligrams per kilogram per day. This was based on findings observed at the 30 milligrams per kilogram per day, including decreased fetal body weight, cervical vertebrae abnormalities. And then here's the finding in question for our analysis was the increased incidences of altered texture of the cut surface of the islands. So on the surface, it looks straightforward. We had a NOEL of 10 milligrams per kilogram per day. But when we looked more closely at the dose response data and the weight of evidence of data, some questions emerged. Next slide. So first, the lens abnormalities showed statistically significant dose responses at all the doses, including the 10 milligrams per kilogram per day. And there were no lens abnormalities in the controls. The study authors had acknowledged that the finding is normally considered a process artifact, but they also said they could not be excluded as a real effect. We consulted our developmental toxicology subject matter experts, and they informed us that lens abnormalities hadn't been classified as a malformation or variation. So we had some uncertainty about its toxicological significance. But then secondly, and most importantly, we had some biological plausibility. That earlier 90-day study showed the cataracts in rats. And so there was, a question if the compound may have a mode of action affecting the eyes. So this raised the question of whether we could establish a NOEL for this study when all the tested doses showed effects of these lens abnormalities at the lowest dose. So we decided that benchmark dose modeling could provide a more refined point of departure by using all the dose response data rather than just being limited to the tested dose levels. Next slide. So we used US EPA's BMDS version 3.2 at the time, which employs Bayesian model averaging, and the model type was nested dichotomous. And as Lynn and Alan explained earlier, the nested dichotomous approach is critical for developmental toxicity data. We're looking at fetuses within litters, and fetuses from the same mother tend to respond more similarly to each other than fetuses from different mothers. And so if we ignored this nested structure and treated each fetus as an independent, we would have gotten biased estimates. So the nested model accounts for this by treating the litter as the experimental unit. So we also included implantation number as a litter-specific covariant to account for the dam's condition prior to dosing. We calculated BMDLs at 2 benchmark response levels. 5% extra risk was our primary value, and then 10% extra risk for comparison. The EPA guidance recommends 10% as the standard for dichotomous data, as it represents the limit of sensitivity for most toxicity studies. However, again, this was a developmental toxicity study that has greater sensitivity due to the nested design. So the 5% extra risk is more appropriate for the developmental study. We used a 90% confidence level, which is standard for regulatory applications. We included implantation number as a litter-specific covariant to account for the dam's condition prior to dosing. And we modeled only the endpoint in question, which was the eye lens cut surface altered texture incidence. And we focused on the lens endpoint because that's where we were needing to validate the NOEL. Next slide. So here's the results. Looking at the table, at 5% extra risk, we obtained a BMDL of 10.07 milligrams per kilogram per day. And then at the 10% extra risk, the BMDL was 21.22 milligrams per kilogram today. And what's notable is that this BMDL is essentially identical to the study's reported NOEL of 10 milligrams per kilogram per day for the developmental toxicity. So this convergence validated that we could confidently use the study NOEL as our point of departure. And despite the statistical significance of the lens finding at all the doses, the BMD analysis, which used all the dose response data, confirmed that 10 milligrams per kilogram today appeared to be an appropriate value. And then additionally, if you notice, the NOEL of 15 milligrams per kilogram per day from the 90-day study actually falls right in the middle of that BMDL range between the 5% and the 10% extra risk. So that provided even further cross-validation across the independent studies. Next slide. And so what are the key takeaways you could apply for this case study? Well, first, recognize when BMD adds value beyond our traditional approaches. We had statistical significance at 10 milligrams per kilogram per day for the lens finding, but the BMD modeling helped us validate this NOEL and ensure that it was still defensible by using all the available dose response data for that endpoint. And then second, match your statistical model to your data structure. So for developmental toxicity studies with fetuses nested within litters, the nested dichotomous model is essential for ensuring to get unbiased results. Third, use cross-validation to build confidence. The BMDL of 10 milligrams per kilogram per day validated the study's reported NOEL, and then both were consistent with the independent 90-day study in rats with the NOEL of 15 milligrams per kilogram per day. So when multiple methods and independent studies converge, that really strengthens your scientific justification for your case. And we used that validated NOEL for our safety assessment and concluded that there were no safety concerns for the current intended uses for that anti-static agent, including the infant exposure. So next slide. If you're considering using BMD modeling in your food contact substance submissions, please just provide clear documentation, like my example today, of your methodology. Include your model selection rationale, your BMR justification, how your results support your safety assessment. And then if you'd like to discuss your BMD approach before your submission, we're happy to take a pre-market consultation. You can find more information about that on our FDA website. So thank you for your kind attention. I'm happy to answer questions during our panel discussion. Thanks.
Thank you so much, Omari and Laura. We really appreciate it. So introduce our last but not least speaker. We have Olivia Osborne from the UK Food Standards Agency, who will be talking about the state of the science using BMD to establish health-based guidance values. Welcome, Olivia. Olivia, we are seeing your screen, but we are not hearing you.
Can you hear me and see me now?
We can hear you. I cannot see you.
I don't think it matters if you can see me or not.
That's okay, yes. And then we do not see your slides in presenter view yet.
Can you see them now?
Perfect.
Perfect. Well, first of all, thank you ever so much for HESI inviting us to present the state of the science. I'll be talking about reviewing the utility of benchmark dose modeling as a chemical risk assessment tool. And this is in the context of I am presenting on behalf of the Committee on Toxicity. But first of all, who are we? So I work for the Food Standards Agency, and our job is to use our expertise and influence so that people can trust that the food that they buy and eat is safe and what it says it is. At the Science, Evidence and Research Division, where I work, this provides strategic analysis, insight and evidence across the FSA's remit to underpin the development of policies, guidance and advice on food safety. Our framework for science government sets out what we do to make sure we live up to our principles of being open, transparent, and based on scientific evidence. Our strategy is food you can trust, and this means that we have 3 pillars, which is food is safe, food is what it says it is, and food is healthier and more sustainable. We have currently 4 research priority areas, and these are areas of research interest. We do review and update these on a regular basis, and this is to ensure that these align with our key evidence gaps. Now, who do we work with? So where is this reviewing state of the science coming from? So this is coming from the Committee on Toxicity, which is who we work with. So the Committee on Toxicity is an independent scientific committee that provides advice to the Food Standards Agency, the Department of Health and Social Care, and other departments on matters concerning the toxicity of chemicals. And their independent expertise and knowledge is vital to the work that we do. And what are we, the FSA, currently doing is that to keep up with the technological and scientific evolutions, we are using the latest technology and best available scientific methodologies to develop and incorporate these additional tools into our regulatory risk assessment portfolio. This is to evaluate chemical safety in food more efficiently without compromising the quality. And what we actually have done is we've published a blueprint roadmap, which essentially is to develop, endorse, and finally towards regulatory acceptance of new approach methodologies in which BMD would be included in this. Within this roadmap, there are 7 steps. It doesn't seem to want to go forward. There we go. And what we've done here is we're going to identify and review research and development and review and recommend, which is exactly what we did at the Committee on Toxicity. So back to reviewing the utility. So what we did was we published this special science and research topics report, and all of the links are available there, and they will be available obviously after this talk. And what the committee then came to some overall conclusions were, which already in the whole workshop I've already overlapped on this many times, but the fact that you can use the entire dose response data set is obviously one of the major advantages of BMD modeling. Unlike the Noel approach, which relies on identifying a single dose with no significant statistical effect, the BMD model can actually fit a mathematical curve to all available data points. So this allows for a much more comprehensive data analysis and reducing the influence of arbitrary dose response selection, and in theory, increasing the reliability of a derived reference point. The flexibility of BMD modeling also is the different types of data, which is also something we've seen in this workshop. The fact that you can actually apply it to a continuous quantal endpoints, and it's suitable for traditional studies, as well as these new emerging types such as gene expression and high throughput screening data, and those new approach methodologies that the last talk alluded to. BMD modeling also facilitates the derivation of reference points. So this is consistent and comparable across different substances. And due to BMDs being linked to the defined effect sizes, they can be used to calculate relative potency factors or toxic equivalent factors, which are essential for assessing chemical mixtures or structurally related compounds. So this, they believed, is very, very useful, especially in cumulative risk assessments and prioritization exercises. So what did they define as the current challenges to the use of benchmark dose modeling in regulatory toxicology? So one of the challenges is the divergence in guidance. So we've seen a lot of guidance as well in this workshop, and the methodological preferences between these different regulatory bodies. Statistical philosophy creates inconsistencies in how BMD results are derived and therefore interpreted. So this could potentially lead to different health-based guidance values for the same substance, depending again on the approach used. Technical challenge also persists in the practical implementation. And actually with the technical challenge, they also included expertise within this. You have to be an expert to be able to actually not only use the BMD modeling software, but actually interpret the results. And talking of software variability adds another layer of complexity. So if there's this variability, it can get quite problematic when you're trying to compare it in regulatory contexts where consistency and reproducibility are absolutely paramount. So within this paper, we reviewed some of the BMD modeling software. So the EPA one that we've discussed, the EFSA one, and BMD Express. And what I'm going to do is actually hand over to the user case. So we have a Food Standards Agency computational toxicology fellow. And what he actually did was use various programs of BMD modeling software. So over to you, Arthur.
Hi, thanks, Olivia. Just a brief comment on what Olivia just said. I'll just clarify this various steps of this work that we've done, and it's already published on Frontiers in Toxicology, called A Novel Method to Derive a Human Safety Limit for BFOA by Gene Expression Profiling and Modeling. In this work, we put together an in silico framework, starting from sourcing transcriptomics data from a study developed by Health Canada, published in 2021, where they exposed liver spheroids to 4 different perfluorinated chemicals. We focused on PFOA just as a proof of concept in this case. And the first layer of benchmark modeling, benchmark dose modeling here, was on step 2, where we just replicated the BMD approach that the study from Health Canada reported. And then we conducted a pathway enrichment analysis, found out what was the most sensitive pathway in there. Then we got the concentration values and started working on the calculation of in vitro free concentrations for PFOA using specific models such as the Armitage model and also the Fisher models. And then we've done a reverse dosimetry approach coupled with a PBPK model that was previously published by George Luizu and his group. And then we generated a virtual population of 3,000 individuals, and therefore we used PROST to get the internal doses that we could derive from the reverse dosimetry approach. and then calculate a benchmark dose modeling for that specific alterations, referring back to the most sensitive pathway, most sensitive gene that we found out in our proof of concept. And then we could come up with a health-based guidance value that is probabilistic and population-based as well as using only new approach methodologies to fully integrate the whole data set. So basically, we used different software to do the benchmark dose modeling here with the specific cutoffs for each of them. And we could come up with a very interesting user experience to provide a very good and report back to the FSA in terms of how interesting it could be to put together something related to the BMD modeling as well from the user perspective. So yeah, this is just a really brief comment on that. Please do access the paper, read it, and go through the details. I'll be happy to discuss about this after the talk. But yeah, just being conscious of time, I'll hand over back to Olivia. Thank you very much, Olivia.
Thank you, Arthur. That's very very succinct. So based on this, we then went back to the Committee on Toxicity, and they came up with some recommendations. So they viewed it as that BMD modeling should be complementary rather than a replacement tool at this current stage. Number 2 was to improve transparency and understanding of BMD models. And this is because sometimes we don't understand the complexities behind the model or the parameters. And I think a lot of people also talked about reporting templates and things like that. So number 3 is to standardize and validate BMD in some capacity. The methodologies through case studies, so you've heard obviously one today from the FSA, but this we believe also will help in BMD confidence. Align the study design with modeling needs, inter-committee harmonization. I think that's also been alluded to as well. And number 7 was very important because we do need to consider the resource implications, not only for our agency, but other government departments as well. And this is obviously coming in through the expertise that are needed as well to interpret some of this data. So some of the good then. So BMD modeling can actually offer a more sophisticated data-driven alternative to traditional NOEL-based approaches in chemical hazard characterization. And this is because it allows for the use of full data responses, quantifies uncertainty, and supports more nuanced establishment of the health-based guidance values. So they do see it as a valuable tool that can actually bridge traditional toxicology and emerging mechanistic approaches. The not so good is that the broader adoption is challenged by these methodological disagreements. There are multiple technical complexities and there are some practical limitations. Now, these key issues include the difference of statistical frameworks, as I discussed earlier, in terms of statistical philosophies, benchmark dose response selection, model averaging. All of these can lead to inconsistent outcomes. So it's this idea that depending on the user views on things, obviously this can also inherently move the model in a particular way. The technical hurdles such as model fitting can create more difficulties, and in future there is the uncertainty of the integration of new data types. So we talked about new approach methodologies, we've talked about obviously gene profiling or high throughput data, but what about new data types? Where's the software variability? And that need for expert interpretation can complicate the implementation. And the growing complexity of these tools also raises the concern about transparency and the risk of black box modeling. So what did they see as the future scope? So they believe that the successful integration into regulatory practice will depend on clear and harmonized guidance, improved training, collaborative efforts to ensure the consistency, transparency, and scientific integrity in its application. And with that, I'm going to finish because I am conscious of time. So thank you ever so much for your listening. And as Arthur said, happy to answer any questions. And if I can't answer them, I'm happy to put them to the Committee on Toxicity on your behalf. Thank you very much.
Thank you so much, Olivia. Appreciate you you and Arthur speaking on this important topic. And I think it was a really nice way to wrap up the talks because you had a lot of the sort of benefits and challenges listed there that I'm hoping we can talk about in the Q&A and the panel discussion. So I'd like to invite the people who were speakers for this last session to turn their cameras on, if you don't mind, because it'll give it more of a panel Q&A type of feel. And there's a lot, I just wanted to highlight that there's a lot of questions in the Q&A. And one of the things that I've noticed over this past 2 days is there's very much a patchwork of questions, some of which are very deep into the specifics of the models and the assumptions. And then there's these kind of high level questions that have come up. And so what I was hoping to maybe touch on in this panel is some of those kind of high level themes that we're hearing over the past 2 days. All that to be said, as was mentioned before, we do plan to try to address most of the questions from the Q&A in writing, and many of you as speakers have already done that already. So I just wanted to kind of bring us back to what the goals of, no problem about cameras being disabled, apologies for that. The goals of this meeting and what we were really trying to do with this is to identify where there might be opportunities to kind of, for lack of a better term, harmonize and bring the different communities together. And what we've heard over these past 2 days, of course, is that many of us, both the people who gave talks as well as the participants, are coming from different angles on this. We're coming from different sectors. We're coming from pharma, cosmetics, food, industrial chemicals, ag chem, different regulatory agencies, whether you're doing pharma regulation, chemicals regulation, also people that are doing research on different endpoints, different study types. So there's it's not very, very uniform. And one thing I wanted to ask those who gave talks in this session is there one key kind of challenge or opportunity? So it could be both a negative or a positive that you've taken from these 2 days of discussions related to benchmark dose that you'd like to highlight or bring up. And I'll let you think for a minute because I didn't tell the panelists that I was going to ask this question. Go ahead, Andreas.
Yeah, maybe there seems to be a tendency to focus on the limitations and of BMD and going down these rabbit holes where you could optimize 6 different models until you get the second decimal right. But in fact, we should embrace the possibilities that are offered by BMD modeling and say emphasize how much better it is over those vintage methods that we have used over decades in toxicology. But to use the whole dose range is so much better. I cannot see why people are so hesitant to use BMD methods, while in fact they're a huge step forward.
Thanks, Andreas. Matt, would you like to respond?
Oh no, I wanted to concur. That in the last talk we heard, like the lack of consistency amongst BMD methods poses a problem. But if I'm understanding the literature correctly, the no AL, low AL type approach, there's 2 orders of magnitude often in repeat experiments. And when we're getting down to, say, I don't know, a 1.1, 1.2 fold difference in our lower bound estimates, we're told that that's a lack of consistency when there historically is 2 orders of magnitude. So I think true, there are black boxes, true, there are things that aren't necessarily understood by all, but if it's not looked as a step forward in this regard because of all the different things happening, I think that, that's kind of it's definitely something that needs to be discussed because a 2-fold magnitude is pretty large.
Thanks, Matt. Amari?
The one thing that as you asked that question, I didn't even think about until you asked the question. And I mentioned NAMs in my presentation and my slides. And I think some of those similar types of challenges that you hear, the hesitancy, this is new, and it's, hey, it's promoted as being able to advance the way we evaluate and assess safety of different chemicals. But you run into that hesitancy within the NAM space. I'm hearing some of those same, challenges, hesitancies as from the standpoint of BMD as well. There is promise, but you still have to get past that hurdle of people being, hesitant and you just have to keep pushing forward. So, that was just something that hit me as soon as when you asked that question about the overlap there and those the challenges in both areas.
Thanks, Amari. And actually, there was a comment, Stephen, in the Q&A wrote the same thing, which is a lot of these themes that we're seeing related to the challenges are coming up across, especially in that NAM space as well. And I think on Andreas and Matt's points related to our current methodologies and the limitations of those, and understanding that, and how we'll call it new approaches, although I wouldn't say BMD is new necessarily. It's been around for a while, but newish. I think that's a really important point. Guangchao.
Yeah, indeed, just to echo what the speaker said, I understand there's some concern, different methodology, and then for some people that may really sound a bit sensational, and let me use this word. But actually, when your data set, you guys don't see me. Oh, I'm sorry.
No, it's
Yeah,
okay.
now
We can.
now you
Now
do.
we can. There we go.
Yeah. So actually, when your data set is rather good quality, and it's quite informative regarding the dose response, with different tools, more or less, you would get actually comparable results. So what we're dealing with before, for instance, some historical data set, and then the quality is bad, and then we need to figure out different ways, priors or model averaging or covariate approach, But now we also have the opportunity to, in the future, to optimize our study. That's why the study design. And when the study is relatively good or improved, I think more or less you can look past a little bit if you want to be practical before reaching a point, when the harmonization reached. So meanwhile, just as a practical thing, as long as, like other people mentioned, be clear about your choices, and then, transparent. That's my point of view.
Thanks, Guangchao. Does anyone else kind of want to weigh in on their kind of key takeaway? Go ahead, Jose.
I just want to say that most of the time the people focus on the bad examples, the examples in which they find the real differences, okay? And this is basically because the information that you are trying to pass to these algorithms are basically very poor. And you are trying to estimate something that you are not able to estimate with the information that you are passing. And we just focus on those examples all the time, just to basically highlight the difference between the method. More advanced methods are not more reliable in a sense, but I think that we should just look at in how many cases they are basically coinciding or basically producing comparable results. And that is the things that we should also advocate more, because we are always focusing on those examples that highlight the issues, but those examples in general, if you just dig in it deep, you see that basically the data sets are failing. It's not the methods, are the data that we are using. Okay.
Thanks, Jose, for that. Anyone else want to get in? I have other questions for you guys, but just wanted to give everyone a chance to see. Go ahead, Laura.
So, like from my own example, you had a notifier who sent in more data, which should have given us more confidence, right? But it brought up a question of whether or not it was a true Noel or not. So if we hadn't used the benchmark dose, there was risk that it would be a low L. You would have added another safety factor potentially, depending how the experts had discussed it. So in this scenario, it utilized animal data to its fullest. It brought about scientific justification. It brought about confidence in this scenario, and it used the full weight of evidence of all the data that was submitted. And we feel confident, at the end of the day, that our ADI is something that's useful for this food contact substance, and useful for looking at cumulative exposures, not just that intended use. And it was for a sensitive population as well, looking at infant exposures. So I think in this case, BMD really supported not only the FDA in being confident, but also the notifier. So, it's been useful in those ways to help tease out some bringing more confidence to our decision making.
Thanks for that, Laura. So I had a question related to that, because I think Jose's point related to focusing on too much on the negative, I guess you could say, the cases where it doesn't work. And I think one of the issues, and Alan, you just put this in the chat, so thank you for that, to the host panelist chat, is related to there's a lot of instances of where this actually has been very beneficial and works well. The cases where maybe it doesn't, what are the underlying reasons for that, or the challenges there? And I think study design has come up a lot, related to not only our existing, say, traditional in vivo studies, but also if you have new methodologies, you have transcriptomic data, you have in vitro data, you have new methods that are coming online. And the question is, what sorts of analyses are going to be done? How can those new studies be designed such that this type of analysis could be used? But then I think the other piece that's brought up many, many times is our existing studies that we rely on still, despite NAMs. The traditional kind of OECD types of test guidelines were not really designed with this type of analysis in mind. And so how do we address that going forward? And I just will say there was one comment, and I know Jose commented on this, that there are discussions at, say, the OECD level, or maybe even others, ICH, about how to potentially update test guidelines to address this question. And I think you mentioned, Jose, there were some ongoing discussions in that space. I don't know if you might be able to elaborate on that further, or if there's other people who've been involved in that who could speak to that at that kind of high level. Do you want to jump in, Andreas, and then we can maybe have Jose speak to that?
I want to comment first on the in vivo guidelines. I mean, these are minimum requirements. Nobody keeps you away from using 5 dose groups instead of 3 dose groups. Just drives up the cost, and it's not exactly 3R friendly. But if you're a sponsor and you want to have excellent data and not repeat your study, which in the end will even use more animals, do one single good study instead of 3 crappy ones. Very easy. And with regard to the, for example, transcriptomics or toxicogenomics data interpretation, please allow me to be very blunt here. There is currently no guideline on that very end, right? You cannot get any guidance how long to treat cells with what top concentration, and then sample at what time point, and take the data to what level and curate them. So why should we regulate the application of BMD for that type of sort of unregulated data before not taking the first step and aligning that part first, and then continue with quantitative interpretation?
Thanks, Andreas. Did anyone want to respond to that? And then we can...
I'd just like to add that, there is an element of 3Rs even to the in vivo gentox quantitative approach, in the sense that you may not have to do CAR-C studies, especially if you start factoring in ECS and other tools for API. You can clearly see that there will be a decrease, and there could be a decrease in animal usage for those kinds of approaches, right? So just wanted to put that in there as well.
Thank you, Bob. Jose, did you want to step in related to kind of some of the ongoing OECD discussions?
I know that OECD is basically revising the guidance on ecotox studies, okay, and the analysis of ecotox studies, and they are touching upon study design as well. I know that they are doing that. They are currently doing that. This is something that is ongoing.
Thank you, Jose. Go ahead, Paul.
Yeah, I think you've touched on something that's obviously important, that there's this tug of war between study designs that are conducive to BMD analysis and study designs that are guideline compliant. And altering the guidelines is a lengthy, difficult process. And Andreas is right that it doesn't, just because the guideline says you have to do 4 doses or whatever it is, 3 dose plus control minimum, it's just a minimum. But it's true that if you want to move towards 3 Rs, you need to somehow rework the way you're designing your study so that you distribute your experimental units, whether they're culture plates or animals, into more dose groups with fewer animals per dose group. And I think that our empirical analysis of our data sets with large numbers of doses and large numbers of animals per dose group shows this consistently, that you can really maximize the utility of your animals if you distribute them to more dose groups, even if it's the same number of animals. So if it's 24, you're better off doing a 6x4 than a 4x6. Anyways, I think you've touched on something that's critically important, and there's, I think guideline revision is probably the way to go, but it's going to take time.
Thanks, Paul. And I see Matt has his.
Okay, I will talk because I'm assuming you gave me the chance.
I just wanted to touch on. I think you said. Oh, go ahead, Matt.
Okay, I missed you, so.
No, my connection just got a little sticky, so I'll let you go ahead and maybe it'll figure itself out.
I just wanted to kind of stress that even if or when the OECD guidelines are updated and there are different guidelines for the experiment, there's still going to be a problem that can happen. It's not going to be removed, it'll be mitigated. If the problem that Jose was talking about is if you miss the dose response or if you have a very little data to inform that dose response, regardless of how the study is designed, if the actual data miss the dose response, you're still going to be in the situation that Jose was talking about, where a benchmark dose itself requires other information, and that's going to be always problematic.
Yeah, we're not going to solve everything full stop, right? Thanks for that, Matt. I did see a point Alexandra put in the chat. Is there a mechanism for HESI and affiliated agencies to develop guidance for study design that supports dose response analysis? And that is one of the things that we're talking about, not just generally, but one of the things within this group. And in the presentation yesterday, Claire kind of teed up this meeting is, can we bring multi-stakeholder groups of people together to talk about the issues and figure out what might be possible going forward? So it's a little bit of a softball question. Thank you for that, Alexandra. But that's kind of what we're trying to do. It's not necessarily one single dose response analysis guidance, but where are there opportunities for us to kind of have these discussions and come up with best practices or principles or something like that going forward? Just monitoring the chat to see, is there anyone else while I take a look at the chat that has any kind of overarching themes that they've had come up that they'd like to highlight? There's quite a bit of sort of very detailed discussions and questions on some of the key approaches and methodologies. I just wanted to highlight we've of course touched on study design, which is, we haven't solved that by any means, but one of the things that we're trying to do within this HESI group is to identify where we can have a deeper dive discussion. So some of the things that have come up about, obviously study design is a big one. We've talked a lot about differences in different tools. And I think one of the things that was really highlighted this morning was kind of this idea of model uncertainty, model averaging, and those kinds of things. So I'm curious if there's anything from the panelists that you might want to raise related to some of the questions that have come up related to whose model do you use? We have Bayesian and frequentist, which I think there's been a lot of discussion related to that. Questions related to, and I know that Jose responded in the chat, into changes in models and making sure that there's transparency. But is there anything that kind of came up related to differences in which approaches are used and which models are applied, and key things that going forward we want to make sure that we take into account? Jose.
I just want to say, I mean, we have, for instance, there are two different Bayesian implementations that have been presented in this workshop. The one that Matt was talking about was ToxicR, and the one that EFSA developed. There is another one that is called BMD that now is paid. Before it was free, which you could also use at the time. Now not anymore. But I mean, all of these have different model sets, different prior distribution that are used for the parameters, different parametrizations. And this is also useful for sensitivity analysis, okay? This is as well is also important. I mean, the diversity doesn't mean that you are, not having harmonization. On the contrary, it just basically allows you to assess if this diversity poses a problem or not. And as you could see from Matt's presentation, the results of the two different Bayesian implementation, even though they have differences in terms of how the BMD was defined, what was the set of models that was used, and how the implementation of the Bayesian framework was basically done, still they produce very comparable results. I think that it's not bad.
Thanks, Jose. Matt, did you want to comment?
Yeah, I wanted to second what Jose was saying. Jose and I were talking several years ago, and he showed me data sets where there were, I think, 4-fold magnitude differences between the frequentist approaches. However, the Bayesian model averaging approaches were like different, but they were so qualitatively different, you would have had the exact same response. Like I remember one specifically that was 15 mgs per kg per day to 15.3 mgs per kg per day. And that was a difference between my software and Jose's software. And I think there's a reason why we've gone Bayesian is to help solve the situations where there's that quite literally 4-fold magnitude. And you do the sensitivity analysis and you do these things, and the extant programs will usually get the same answer. It's because there's only so much information you have, but if you also think of it, there's only so much curvature a dose response curve can have. You don't expect to go from 0 to 20% risk with 5 molecules. So those types of dose responses are ruled out a priori, usually in the Bayesian sense, and you get these very different things. So like as Jose said, there's talk about harmonizing the approaches, but I think what we did with the World Health Organization and EFSA and EPA, they're really all getting to be about the same qualitative answer nowadays.
So a follow-up question to that, and I know there's been a bit of discussion. So if we just take the Bayesian approaches, right, and you have, like you said, they're not, in many cases, not that different. How do you rectify the fact that we don't have to pick one model to rule them all, right? We don't have to just get consensus on this is it, with the fact that they're, and having an infinite number of choices, even if they're pretty similar, causes a lot of confusion and barrier to uptake. So how as a scientific community can we provide clarity on that for the people who are going to be having to use this? Again, there's sort of that balance between where science is, there's new evolving science, there's multiple approaches and models that can be done, weight of evidence, but there's also the reality. So I'm curious your thoughts on that.
Well, I can throw mine first out. It's just the simple fact that a priori, there's an infinite number of models, all the dose response models. If we knew what the dose response model was to begin with, then we would know what to fit and there would be no uncertainty. So I think that the problem is this is such a multidisciplinary field where I, as the statistician, the mathematician, have to trust the toxicologist that the data are correct to get to that point. And then there has to be a handoff where you all have to trust us that we're doing what we need to be doing. That's the best way to do it. And I think a lot of these issues and these conversations I've had over the years come to the fact that most people don't like statistics in math. And thus, when you go at the higher level, the whole trust me, it works doesn't necessarily, there's not a lot, the buy-in goes down just because of that whole black box approach.
Thanks, Matt.
Yeah.
Go ahead, Jose.
Yeah. We have tried to be a bit more transparent to publish a technical report with all the information about the framework that you could dig into the math if you want to. Of course, it's difficult. I understand that. And it's true that having many different options doesn't make your life easier. But before we were having PROS and BMDS. We were having two options anyway. As the options have been always there. And before there was also different options in how to handle specific data sets in which you constrain the D parameter or not constrain the D parameter. Now this has been handled now in a different way. It's not in the same way because we are not using exactly the same priors, but still deriving very similar results when the data sets are rich enough to be able to estimate the BMD with the level of accuracy that you would like to. Most of the time we are focusing still on those examples in which the information is scarce, and there we just derive different results, okay? But we shouldn't be focused on that. I mean, we should be questioning, should we analyze this data? Should we use this data to set health-based guidance values? This is the question that we should pose, not questioning the methodology, because if the data are rich enough, methodologies are providing you qualitatively the same results. That's why are we questioning the methodology when it is for a specific data set that the methodology produce different results? It's the data that we should just question, because there are assumptions that we are making in our methodology. All methodology are used. I mean, statistics in general, this is just an application of specific models in statistics. In general, they all make assumptions, and we always make assumptions. And all these assumptions are basically as well embedded in the conclusions that we draw. Thus, I think that, we always blame the methods, but we never look at the data. And we question the data. And we question our understanding about the data.
Thanks, Jose.
And that I think that is something that we should do more.
Andrew, you want to jump in?
Yeah, absolutely. I want to kind of echo what Jose just said. There was an example that I was working with Matt Meyer and Annette Dodge when we were looking at the benchmark dose modeling implemented in Mute-CCAR. And we played with ToxicR with the EPA and the EFSA different priors, and we compared it to Prost. And again, I think it comes down to we got one edge case, which looked a little scary, but I think it really came down to it was data that shouldn't have been modeled, where you don't actually have a dose response. So Prost basically said, don't model this, and ToxicR provided estimates. One on one set of priors, which looked very, very certain, and the other set of priors showed you a very wide credible interval, which would say there's nothing there. But again, it's one of those things where, like Jose said earlier, we base these decisions on these edge cases. Like when I saw that, I got a little scared. But it's true. You got to look at it on a body of data sets. If you look at like 100 data sets, everything looks great. But that one data set scared me. So again, it's, and moving into something new. But I just wanted to kind of highlight that and echo Jose's comments.
Thank you, Andrew. So we're getting towards the end. I just wanted to highlight, I think much of this is to try to kind of, we'll call it thread the needle between, of course, the point about we need to really look at the data and figure out what should be done and what the science is telling us. At the same time, you have practical limitations related to the fact that you need regulatory review of things and data to be generated and chemicals to be registered or not registered, whatever that looks like. And what we're trying to do is to find a path through that forest to figure out how we can come together as a scientific community, multiple sectors, stakeholders. A lot of this is the fact that we really need to make sure that we're communicating what BMD is, what the tools are, and to try to come up with practical next steps going forward. So this is just the beginning of a conversation. I'm going to turn it over to my colleague Rachel Puglisi to wrap us up. And thank you very much to all our panelists for that discussion.
Great, thanks, Michelle. And yes, thank you everyone who spoke today and to our panelists for that informal Q&A there. So to close out, similar to yesterday, we do have a closing poll. If you have a few minutes to fill that out for us, it's really informative as we kind of move into our next steps and helps us make these webinars better and more informative going forward. So just to wrap things up, I want to take a moment to touch on what we've heard the past few days. What stands out is that despite the diversity of sectors represented here, there are significant areas of alignment. We've seen that benchmark dose modeling is being applied across regulatory sectors, industry, as well as in research settings. We also are starting to see an increased use of using BMD to support risk assessment and decision making. At the same time, there are many challenges, as we've discussed, especially today, across the sectors. There's quite a bit of challenge in navigating the variability in models, software, parameter selection, as well as questions around how to improve data quality and study design. I think we'll need to continue to have discussions on these things as well as we continue to discuss today, how to best balance statistics and biological relevance in interpreting these results. Alongside all these challenges, though, I think there have been many areas of consensus, primarily the recognition of the need for greater harmonization and alignment on identifying key priority areas that need to be addressed. In terms of what comes next, there will be a post-meeting survey coming in the next week or so. So keep an eye on that. It should just take a few minutes to keep out. And again, these are just really helpful for us in planning next steps and planning future webinars. We are planning to keep these conversations going within HESI. This wasn't meant to just be a one-off conversation, as Michelle stated. We will continue to talk through these challenges and identify the priority areas. We'll continue to have discussions and explore where we might be able to form a working group or committee within HESI. So again, just a starting point, and we really look forward to continuing to have these conversations with you all. Just a big thank you to all of our speakers and panelists, as well as those of you who tuned in the 2 days to learn. And we, again, encourage you to fill out our post-meeting survey here. And if you have any questions, you can find us at hesiglobal.org and reach out from there. Right. With that, thank you, everyone, and look forward to continuing these discussions.